Welcome to Science with Tal. In today's video, we will go over the organization and the main anatomical structures of the central nervous system. As a quick message before we get into the video, I want to sincerely thank each and every one of you that has subscribed to the channel and has given feedback on the videos. It is really appreciated and it will certainly help me make more content. Also, for the discussion, I have made a ton of diagrams to illustrate the anatomy of the spinal cord, brainstem, and forebrain, so I compiled them into a PDF that you can consult from the link in the description. If there are some mistakes in this video, I will adjust the cards on the PDF and make a note in the description to keep you all up to date. Now, as I said, the topic of today's video is the anatomy of the central nervous system, or simply CNS. To understand the anatomy of the CNS, I believe that it is first important to understand the general structure of the nervous system to get an idea of where the CNS falls in it. It turns out that the central nervous system is just one of the two main branches of the nervous system. If we consider a general model of the human being, the nervous system is divided into the central nervous system, which is composed of the brain, the brainstem, and the spinal cord. The second division is the peripheral nervous system, and it is essentially made up of the nerves that go in and out of the CNS. There are two general types of nerves that we will come across in this discussion. The first are spinal nerves which go in and out of the spinal cord, and the second type are the cranial nerves that go in and out of the brain and brainstem. Generally speaking, the nerves that go towards the CNS carry sensory information, and they are said to be afferent nerves. Afferent broadly means that it goes towards something, and in this case, it is the CNS. Conversely, the nerves that come out of the CNS generally carry motor information and are called efferent nerves. The afferent-efferent terminology is important to understand as it is commonly used in textbooks to describe the type of information that nerves carry. In addition to this distinction between the nerves, the peripheral nervous system can be further divided into two divisions: the autonomic nervous system and the somatic nervous system. The somatic nervous system informs us about the sensations from our body, and those include the senses of touch, pain, temperature, and proprioception. The somatic nervous system is also responsible for controlling our voluntary movements that are carried out by our striated muscles. An example of this would be the contraction of our biceps. The autonomic nervous system controls, or at the very least, it influences our involuntary functions like our heart rate, respiration rate, digestive activity, and so on. And to mediate these functions, it will often activate smooth muscles and glands that are a part of our internal structures. In both cases, you will notice that the involuntary and voluntary functions include sensory and motor components. Now, the autonomic nervous system can be further divided into three branches: the sympathetic, parasympathetic, and enteric branch. The sympathetic branch is often referred to as the one that mediates our fight or flight response because it inhibits digestion and facilitates movement by making our body pump more blood and breathe more frequently. On the other hand, the parasympathetic branch is often said to mediate our rest and digest because it promotes digestion and counteracts the effects of the sympathetic branch. Finally, the enteric branch is a separate and independent nervous system that locally mediates digestive activity in the gastrointestinal tract. What remains to be added to our picture of the nervous system is the role of the central nervous system, which, in contrast to the other branches, has more of a vague description due to the extent of its functions, and also because of our lack of knowledge about how it operates. In a few words, I would describe the central nervous system as the system that mediates cognitive and reflexive processing of the environment. In other words, the central nervous system mediates cognitive functions such as our memories, our emotions, our capacity to understand languages, our decision-making, and many others that allow us to understand and interact with our environments. I include that the central nervous system mediates reflexive processing because it has, especially in the spinal cord, circuitry that mediates sensory motor reflexes, such as walking to an extent, or the classical example of the knee-jerk reflex. Although these types of processing are not always dependent on our cognition, they are important for our survival and integrate information from the peripheral nervous system. What we have just described here can be considered as the structural model of the nervous system, and it gives a good overview of the different components and their hierarchy, but I believe that this schematic does not fully paint the picture of how the nervous system functions. I have hinted already at the fact that, in the most reductive way possible, The nervous system mediates our perceptions and interactions with the environment. Hence, to understand the functional model of the nervous system, let's consider what our environment is generally made of. As you are probably aware, the environment outside of our bodies is made out of a variety of sensory stimuli that we need to be receptive to survive. These sensory stimuli include light that we perceive with our eyes, sounds that we perceive with our ears, chemicals that we perceive with our noses or mouths, and any form of touch that we perceive with our bodies. These constitute the 5 basic senses that we are most familiar with. Accordingly, a portion of the nervous system called the sensory nervous system is dedicated in sensing these signals and sending them to the CNS for us to perceive them. As mentioned earlier, sensory signals are also referred to as afferent because they go towards the central nervous system. Hence, the sensory nervous system, which mostly originates and travels through the peripheral nervous system, goes towards the CNS. In terms of neurons and synapses, the CNS probably constitutes the largest functional portion of the nervous system because it contains the structures and circuits that interpret the signal that it receives from the periphery. In addition to the perception of our environment, the CNS contains the circuits that allow us to interact with our environment through the movement of our motor system. These constitute the efferent functions that originate in the CNS and end up in the periphery to activate muscles. One element that is sometimes overlooked in the functional model of the nervous system is the fact that the nervous system also mediates the regulation and balance of our internal environment, also known as our homeostasis. The nervous system has a great influence on our internal structures such as the heart, lungs, kidneys, and digestive organs, and to exert its influence, the nervous system has multiple afferent and efferent pathways that are linked with our internal structures. These pathways are mostly carried out by sympathetic, parasympathetic, and enteric branches of the nervous system. An important point to consider in the functional model is that the efferent actions will affect and change the environment, so the CNS has to constantly update its perception to produce accurate actions. The final thing that I want to mention is to keep in mind that these 2 models of the nervous system are highly reductive and obviously require many more elements that I will add in due time to fully understand how the nervous system operates. Nonetheless, now that we have a good intuition on the structures and function of the nervous system, we can start discussing the anatomy of the central nervous system. A critical element that we need to establish before we actually look at CNS structures is to define the terminology that will be used to navigate across the CNS. Whether it is the brain or the spinal cord, because the central nervous system is a 3-dimensional structure, it is generally analyzed through the perspective of 3 axes: up, down, front, back, left, and right. Obviously, up, down, and so on are not the words that are used, and the actual terminology is a bit more complex because the words at first might seem foreign. Nonetheless, let's start slowly by considering the front, back, and up-down axes. Now, generally speaking, vertebrate animals generally fall in 2 categories based on the way their nervous system is constructed. On one hand, there are animals like cats that have a nervous system organized in such a way that the front of their brain follows the progression of their spinal cord, and the top of their brains also corresponds to the top of their spinal cord. For that reason, animals like cats have one set of axes that are consistent across their nervous system. On the other hand, there are animals like us, humans, that have brains that are tilted with respect to the front of the spinal cord. Indeed, if we consider the front direction of the spinal cord like we did for the cat, you will notice that the front of the spinal cord is not in parallel with the front of the brain, and the same goes for the top and bottom axis. As a result, an important anatomical characteristic of the central nervous system in humans is that it is divided into 2 distinct sets of axes, one for the brain and one for the spinal cord. Here, the red arrows represent the front direction, and you see that they point in different ways depending on if you consider the brain or the spinal cord. As you will see shortly, these 2 different sets of axes are important to consider to make sure our navigation terminology is consistent across all organizations of the nervous system. Now, since the cat has an easier nervous system to navigate around, let's start with it to establish the true navigational terminology. The front direction is known as rostral, as it means towards the nose, and the back direction is known as caudal, as it means towards the tail. In terms of up and down directions, Up is known as dorsal and down is known as ventral. If you are familiar with French, you will notice that ventral is similar to the word ventre, which means belly, and as you can see, the ventral direction points towards the belly. Similarly, the word dorsal is similar to the word dos, which means the back, and as you can see, the dorsal direction does indeed point towards the back. Now, when we consider the two axes of the human CNS, this is how it would look. You will notice that at the junction of the spinal cord and the brain, the axes do a 90-degree rotation to adjust for our posture. An important point to mention is that for the axes of the brain, the names generally change, although it is common to see dorsal and ventral being used. Rostral and caudal are a bit less common. So, for up in the brain, the word superior is generally used instead of dorsal, and the word inferior is generally used instead of ventral. Instead of the rostral-caudal axis, the front and back axis is often described in terms of anterior and posterior, with anterior being towards the front and posterior being towards the back. Back to our three axes, you will notice that I have not mentioned the left and right axis, and it turns out that no anatomical structure are referred to as being left and right in neuroanatomy. This is because our bodies and by association the CNS are bilateral. As you can see, the brain has two hemispheres, and if we were to cut the brain in half like this, each hemisphere would mirror the other the same way our right hand mirrors our left hand and our right eye mirrors our left eye. Accordingly, it causes the structures of the central nervous system to have a central axis called the midline, which you can consider as being the plane that divides each side. As a result, if you move away from the midline, you go in the lateral direction. Since both sides are pretty much mirror copies of the other side, the directionality between left and right doesn't really matter anymore. In summary, we get this system to navigate the CNS. This summarizes pretty well what we have discussed, but there is an important detail that remains to be mentioned, and it concerns how this system is applied to the brain and the spinal cord respectively. So, as we mentioned for the brain, up is superior, back is posterior, down is inferior, and forward is anterior. In addition, some of the nomenclature for brain structures will use the ventral and dorsal terminology, and because of their definitions of being towards the top and bottom, their axis when applied to the brain will go in the same direction as the superior and inferior axis. For the other axis, rostral and caudal are not often used in the brain, and especially to name structures, so these 6 words describe pretty well how to navigate the brain. When it comes to the spinal cord, you will see shortly that the terms do not all match with our shorthand guide on the right. From what we established, up is dorsal, back is caudal, down is ventral, and forward is rostral. In addition to that, some structures in the spinal cord use the anterior and posterior terminology, which from their definitions of being towards the front and back, as we can see here, it matches up with the dorsal and ventral axis. So, to sort of reiterate the main point that I'm trying to convey here, is that the guide you see on the right matches with the directions of the brain, but not necessarily for the spinal cord. And that is an important aspect to consider when we will discuss the anatomy of the spinal cord. As long as you keep in mind that you are navigating the brain, the guide on the right is safe to use, but otherwise for the spinal cord, it is better to refer to the one in the bottom left. Now, to make sure this anatomical system makes sense, I want to review a few quick examples to show how these anatomical axes are used in neuroanatomy. For example, we will consider the lateral surface of the brain and the medial surface. On these two surfaces, we will look at the frontal lobe here shown in red. Within this region, there is a region called the prefrontal cortex that contains multiple functional regions. Now, the divisions for the prefrontal cortex are a bit abstract, and to make them I followed this general model shown in this study that discusses the frontal lobe. Anyhow, the prefrontal cortex is divided in different regions that are named based on the navigation system we covered. On the lateral surface, the top portion is called the dorsal lateral prefrontal cortex because it is on the dorsal side and on the lateral surface. By the same logic, the region below in green is the ventral lateral prefrontal cortex, and the region in blue is the lateral frontal polar cortex. On the medial surface, the top region will be called the dorsal medial prefrontal cortex, the bottom in green the ventral medial prefrontal cortex, and the last region in blue is the medial frontopolar cortex. The actual names of the regions are not so important, but I hope this example has allowed to demystify some confusions if there were any. Now, as you might have noticed, although the brain and spinal cord have some features that we can examine on their surface, there is a lot of important structures in the CNS that can only be seen through cross-sections. Due to the complex 3D structure of the brain, there are 3 different types of cross-sections that can be made. One from the side or lateral view, one from the top or dorsal view, and one from the front or anterior view. If the brain is cut from the side view, this is said to be the sagittal plane. A cut while viewing from the top would show the horizontal plane, and a cut at the front is called the coronal plane. These 3 different types of cuts provide various angles to analyze the brain and decompose the complex 3D structure in more manageable perspectives. To see all 3 planes on the same brain, here's how it would look. The sagittal plane cuts like this, the horizontal plane like this, and the coronal plane like this. An important application of these planes can be seen in typical MRI scans, where the brain is shown under the horizontal, coronal, and sagittal planes. Hence, understanding how these planes are obtained can definitively help make sense of what each cross-section depicts. In summary, the key aspects to understand how to properly navigate the central nervous system are the 3 axes for the brain and spinal cord, as well as the 3 planes that generally divide the brain. Alright, let's get back to the structure of the CNS. With the neuroanatomical navigation terminology now in our toolbox, we can start exploring the anatomy of the central nervous system by considering its basic subdivisions. This will allow us to set the groundwork for what we will cover throughout this video. If we consider, again, our side view of the CNS, we have already mentioned that it is made out of the brain, the brainstem, and the spinal cord. As you might suspect, all these structures can be divided into many more. One common type of classification for CNS structures comes from embryology, which is a branch of biology that deals with how living organisms develop. Now, as a quick note, I want to mention that I won't dive into the development of the brain in this video, but I'll use the embryological terminology to describe the CNS. For that reason, if these terms that I will use seem foreign, I would suggest consulting an additional resource to learn more about where these words come from to make sure there is no confusion. With this being said, let me show you quickly how early brain development roughly looks like. Given that the CNS comes essentially from the differentiation of a tube, at around 40 days of development, the CNS looks like this. Then, as it continues to differentiate, it progressively shapes itself to the grown structure we are starting to get familiar with. Throughout this development, the early structures that we see in days 40 serve as cues to divide the CNS. There are generally 4 main divisions. At the rostral end of the CNS, in other words, at the very front of the CNS, is a division called the prosencephalon, which also stands for the forebrain or front brain. When we go more caudally to the prosencephalon, we get the mesencephalon, which is also known as the midbrain. Even more caudally, we get the rhombencephalon, also known as the hindbrain, and finally, caudal to these 3 divisions, we get the spinal cord. Now, these 4 divisions can be further subdivided into more divisions that will then be used to describe which structures they become. As a quick note, I will mention and show the important regions each divisions become, but keep in mind that all of these regions will be covered in much more detail later, so I won't discuss their functions at the moment. I simply want to introduce their names and locations so it is easier for us to know what we are talking about later when we encounter them. Anyhow, starting with the prosencephalon, it can be divided into the telencephalon and the diencephalon, with the telencephalon being the more rostral division. In terms of what it will become later, the telencephalon will become the cortex, which is the outer layer of the brain. It will also become most of the subcortical nuclei that are within the brain, such as the basal ganglia, amygdala, and basal forebrain. The diencephalon, which is more caudal than the telencephalon, gives rise to the thalamus, the hypothalamus, and the retina. Following that, we get the mesencephalon, which in contrast to the prosencephalon and the rhombencephalon, does not divide further. It becomes the midbrain, which is a structure of the brainstem that holds a variety of different nuclei, including the superior and inferior colliculus, the red nucleus, and the substantia nigra. Caudal to the midbrain, the rhombencephalon becomes the metencephalon and myelencephalon. The metencephalon later becomes the cerebellum and the pons. The myelencephalon later becomes the medulla, which is also known as the medulla oblongata. Finally, we have the spinal cord, which remains the spinal cord even at later stages. One thing about this embryologic terminology that I want to clarify is the use of the word brainstem. So, although it doesn't align quite well with the embryological analogs, the brainstem corresponds to the association between the midbrain, the pons, and the medulla oblongata. I will often use the word brainstem, so make sure there is no confusion between this word and the other terminology that I have shown you. Alright, now that we have established the main components of the CNS, we can start exploring each structure in greater detail, starting with the spinal cord. So, if you recall our discussion on the functional and structural model of the central nervous system, you will remember that the spinal cord represents an important hub in receiving sensory afferent information such as touch, pain, temperature, and proprioception. The spinal cord is also an important hub for sending motor efferent information to the body via the peripheral nervous system. Recall that the peripheral nervous system mostly communicated with the spinal cord through what are called spinal nerves. Additionally, the spinal cord plays an integral role in mediating many sensory motor reflexes that are crucial to our survival. For that reason, to understand how the spinal cord operates and what each of its components does, it is crucial to understand how the sensory and motor information is transmitted across that structure. But given that the objective of this video is to cover the anatomy of the spinal cord, I will not dive deep into what happens at the periphery, but rather keep it at an intuitive level so we can focus on the anatomy here. The details regarding what happens in the peripheral nervous system with respect to touch, pain, muscle contraction, and so on will be covered in other videos that will focus on these topics. With this being said, let's start from the classic lateral view we are used to see. The first essential element that should be mentioned is that obviously, the spinal cord and the brain are not unprotected like this in our bodies. As you might know, the brain is protected by the skull, and the spinal cord is protected by a complex of bones, muscle, and tendons called the vertebral column. If we take a closer look at a segment of the vertebral column, you will first notice the bones that surround the spinal cord. These bones are called vertebrae, and for our purposes, given that we want to focus on the spinal cord, the main purpose of these bones is to protect the spinal cord. As a reminder, the spinal cord that we see in the middle has generally three important functions, namely, through mechanisms we will cover later, the spinal cord constitutes an intermediate for sensory and motor functions such as touch, pain, temperature, proprioception, and muscle activation, as well as homeostatic functions in the case of the autonomic nervous system. Moreover, the spinal cord contains sensory motor reflexes that allow us to swiftly interact with our environment. The spinal cord also contains what are called central pattern generators, which are circuits that produce rhythmic movements such as walking. To communicate with the rest of the body, the spinal cord is connected with the peripheral nervous system via the spinal nerves that bring sensory afferent information and send efferent motor commands. As you can notice, the vertebrae are made in such a way to have holes, so the spinal nerves can enter and leave the spinal cord. You will also notice from this figure that the spinal cord is highly bilateral. This means that when we divide our bodies in half, the two halves of the spinal cord mirror each other, and that also goes for the spinal nerves that go in and out of it. For that reason, the spinal nerves are usually talked about in pairs, and it turns out that we have 31 pairs of spinal nerves. To see it more clearly, let's go back to our lateral view. If we consider simply our central nervous system and a partial view of the vertebral column, you will see that when we include the spinal nerves leaving the spinal cord, we have 31 pairs, so 62 spinal nerves in total. These 31 pairs of spinal nerves, whether they leave or come from a certain region of the body, supply the sensory motor information for a specific area of the body. Generally speaking, if we consider a front and back view of what the 31 pairs supply, they cover the vast majority of our bodies, with the exception of the front of our faces that is covered by a particular cranial nerve that we will cover later. Now, within this large area covered by the spinal nerves, it is possible to further divide that area based on the region that each spinal nerve pair covers. This given region that a pair of spinal nerve covers, or innervates, is called a dermatome. Consequently, we can say that we have 31 different dermatomes. An important aspect about the anatomy of the vertebral column that I have yet to mention is the fact that the spinal cord is divided into 5 distinct regions called cervical, thoracic, lumbar, sacral, and coccygeal. The names of these regions are based on the location of the vertebra near the spinal nerve. The cervical section, which by the way comes from the Latin word cervix, that means neck, represents the neck region. In this cervical region, it contains 8 of the 31 spinal nerve pairs. The shorthand terminology to classify these spinal nerves is to generally call them C1 to C8 to refer to the spinal nerve pair. In terms of the dermatome region, you will notice that the 8 pairs of the cervical region altogether cover the neck region and a good portion of the arms. The following section, thoracic, represents the chest region and it has 12 of the 31 spinal nerve pairs. In a similar fashion to the labeling of the cervical region, these 12 pairs of nerves are referred from T1 to T12. As you can see from the dermatome regions, the thoracic sections generally cover the upper back, the majority of the torso, and a portion of the arms. Next, the lumbar section of the vertebral column covers the lower back and has 5 of the 31 spinal nerve pairs. The dermatome regions, which are labeled L1 to L5, cover mostly the legs and the lower back. Then, the sacral region has 5 of the 31 pairs and covers mostly the legs. The dermatomes and spinal nerve pairs are labeled from S1 to S5. Finally, the coccygeal region is associated with one single pair and covers the region of the coccyx. 2 important aspects to consider with dermatomes is that first, the regions that they cover overlap greatly, and for that reason, if you have some damage to a particular spinal nerve pair, you won't completely lose sense of touch or movement in that theoretical region. Secondly, the dermatome regions vary greatly among individuals, so it is important to keep that in mind when consulting that graphic. With these regions now covered, there is an important anatomical element that we can now appreciate. Indeed, when we consider a view of the back of the spinal cord, or simply a dorsal view, you will notice that it is enlarged in the upper and lower regions of the spinal cord. These 2 enlargements are respectively called the cervical enlargement and the lumbosacral enlargement. The 2 are caused by the fact that our arms and legs supply and demand more processing from the nervous system, which causes our spinal cord to be enlarged. If we consider backward divisions, you will see that the cervical enlargement occurs between C3 and T1, and the lumbosacral enlargement occurs between L1 and S2. Now, another anatomical element that you might have noticed in these drawings is that the spinal cord is actually shorter than the vertebral column. Indeed, the spinal cord, which, starting from the caudal end of the brainstem, only reaches the first few vertebrae of the lumbar divisions. This terminal end is called the conus medullaris. As a result, the spinal nerves that come from the legs produce what is known as the cauda equina, which is basically the collection of spinal nerves that innervate the lower body and leave from the end of the spinal cord. The final anatomical detail that I believe is relevant to mention is that when we consider the skull, there is a hole in the skull called the foramen magnum that allows the spinal cord to pass through and communicate with the rest of the central nervous system. As you can see, these diagrams are packed with all sorts of information, so if it is the first time you see all these words, make sure to go slowly through them to avoid any confusion. I hope that this final view of what we discussed will help summarize the key components of the external anatomy of the spinal cord. Alright, with this general anatomy of the external surface of the spinal cord now taken care of, let's take a closer look at the spinal cord without any vertebrae to see how spinal nerves go in and out of the structure. This will allow us to establish some important terminology before we discuss the internal anatomy of the spinal cord. Again, we have our spinal cord in the middle, and now you can see more clearly what the spinal nerves look like. In this diagram, we can see 2 pairs of spinal nerves, and as a reminder, the spinal cord had 3 important functions, namely, being the sensory motor intermediate between the body and the brain, having the sensory motor reflex circuitry, and the central pattern generators that produce rhythmic movements. Before we dive into some of the terminology, I want to quickly review the neuroanatomical navigation terms because a lot of the terminology that we will see is based on their logic. So, for the spinal cord, the direction towards the brain is rostral, and the direction away is caudal. On the other axis, towards the back is dorsal, and towards the belly is ventral. Additionally, if we consider this axis of front to back for the body, the dorsal side is also known as posterior, and the ventral side is also known as anterior. The important axis that we will consider for our purposes here is this one, the dorsal to ventral or posterior to anterior axis. In the diagram that we have right now, this side of the spinal cord that is closest is on the ventral or anterior side, so near the belly, and it goes towards the dorsal or posterior side. Because of that, if we divide in the middle like this, everything near us will be suffixed by ventral and away by dorsal. Generally speaking, any sort of cross-section of the spinal cord will generally be oriented like this, with the ventral side closest to you and the dorsal side being the furthest. Now, keeping that in mind, let's now consider the spinal nerves in more detail. As I have said many times, spinal nerves carry both afferent sensory information towards the spinal cord and efferent motor information away from the spinal cord. Starting with afferent sensory information, when it travels towards the spinal cord, it reaches a structure called the dorsal root ganglion, which is an enlarged structure that contains the cell bodies, or cell somas, of the pseudo-unipolar sensory neurons, here shown in blue by the filled circle. After crossing the dorsal root ganglion, the structure branches into what are called dorsal rootlets, here highlighted in yellow, and these rootlets end up conducting the afferents into the spinal cord. When we consider efferent motor information, the cell bodies of the motor neurons originate in the spinal cord and leave by ventral rootlets to reach a structure known as the ventral root before joining sensory afferents in the spinal nerve. Because the spinal nerve ends up carrying both afferent and efferent information, it is also referred to as a mixed nerve. In this figure, you might have noticed that the spinal nerve splits in two. The dorsal portion is called the dorsal ramus, and the ventral portion is called the ventral ramus. Although both rami are spinal nerves and carry sensory motor information, the main difference between the two is that the dorsal ramus carries this information to supply the posterior trunk and the posterior head, and the ventral ramus carries this information to supply the limbs and the interior portion of the trunk. One important element that I hope you have noticed is that sensory information is segregated from motor information as it goes dorsally and motor information leaves ventrally. This will be really important to keep in mind when we will cover the internal anatomy of the spinal cord. Now, what I just told you and what you see in this diagram is only part of the story when it comes to the spinal nerves. Indeed, the structures that you see here only hold for the functioning of the somatic nervous system. Remember that along with the somatic nervous system that controls voluntary functions, the peripheral nervous system is also made out of the autonomic nervous system that controls involuntary functions also with the aid of spinal nerves. However, the autonomic nervous system is much more complex with respect to the spinal cord because there are two different branches, namely the sympathetic and parasympathetic nervous systems, and they both use different mechanisms to reach their targets. As a result, there is much more diversity and exceptions to consider when discussing how the autonomic nervous system uses spinal nerves. So, although I plan on covering the principles of the sympathetic and parasympathetic nervous system in other videos, let me show you a few structures and discuss some key concepts that should be helpful to get an intuition. First, it is important to establish that the main difference between the somatic and autonomic nervous system when it comes to the spinal cord has to do with the processing of the efferent motor command. Indeed, when we consider a cross-section of the spinal cord, motor neurons from the somatic nervous system will leave through the ventral root and go on to directly synapse with striated muscle fibers for voluntary control. On the other hand, the sympathetic and parasympathetic branches use intermediate ganglions before reaching their targets, such as the heart. As we defined previously, remember that a ganglion is a term used to describe a collection of cell bodies. As such, if we first consider the sympathetic nervous system, the efferent commands that will leave the spinal cord will leave by the ventral root and contact a ganglion where they will synapse on another neuron that will then reach the target. Because of this ganglion intermediate, the first neuron is usually referred to as a preganglionic neuron and the second as a postganglionic neuron. When it comes to the parasympathetic nervous system, it somewhat has the same logic with the ganglion, but the preganglionic neuron originates from a different part of the body, for example the brainstem. Now, this is a very simplified view and a broad generalization of what happens with the autonomic nervous system, as there is much more to consider in terms of the targets, the neurotransmitter released, what activates the preganglionic fibers, and so on. For our purposes, the main difference when it comes to spinal cord anatomy that we will consider is the location of the ganglion, because as you can see here, the ganglions for the sympathetic nervous system are very close to the spinal cord, whereas they are further away for the parasympathetic nervous system. Back to the schematic, let's take a look at how the ganglions are structured for the sympathetic nervous system. So, it turns out that the sympathetic ganglions are organized in this structure called the sympathetic chain ganglia, or sometimes called the paravertebral ganglia. As you can notice from the name and the figure, this structure is essentially multiple ganglions connected to one another. The length of the structure extends from segments T1 to L3, so it essentially covers most of the distance of the vertebral column. As we mentioned previously, these ganglions contain the cell bodies of postganglionic neurons that go on to reach their respective targets. To communicate with the chain of ganglia, the efferent preganglionic neurons enter the chain through a small pathway called the white communicating ramus to synapse on postganglionic neurons that then leave the chain ganglia to reach the spinal nerve through the gray communicating ramus. The reason why one of these pathways is called gray and the other white is because the gray communicating ramus is unmyelinated and thus appears darker in imaging studies when compared to the white communicating ramus. Now, because of the chain-like structure of these ganglions, the preganglionic fibers have the possibility to move up and down this sympathetic trunk to synapse at postganglionic neurons that are responsible for different segments. Moreover, preganglionic fibers also have the capacity to continue their journey through nerves called splanchnic nerves and reach more distant ganglions called prevertebral or collateral ganglions. Now, keep in mind that this structure is simply for the sympathetic nervous system, and the parasympathetic nervous system uses different pathways to reach its targets. Indeed, apart from the preganglionic fibers that arise from the brainstem, the segments S2 to S4 in the spinal cord have preganglionic fibers that leave the ventral root, and by way of the pelvic splanchnic nerve, they reach their postganglionic target. In summary, the nerves of the somatic nervous system compare like this to the nerves of the autonomic nervous system. The two models share many similarities and also differ on a few aspects, but nonetheless, this gives us a good overview of how the closed structures outside the spinal cord are organized. Again, these diagrams are very packed with information, so make sure to go slowly if it is the first time you see these words. Alright, with these structures now clarified, let's now consider the internal anatomy of the spinal cord. To do so, let's take a look at a cross-section from the cervical, thoracic, lumbar, and sacral divisions of the spinal cord. The first glaring aspect that you might notice from these cross-sections is that regardless of the level, they all pretty much look the same, and indeed, it turns out that although they have small differences between the regions, they generally share the same anatomical and functional properties. This fact makes sense because as a processing unit, the spinal cord is very stereotypical and processes sensory and motor information somewhat in the same way between the arms, the legs, the torso, and so on. For that reason, we will cover the internal anatomy of the spinal cord by considering a general cross-section that will apply for all. One important difference that I want to point out right now, before we get into the general cross-section, is about the size of the cross-sections. As you can see, relative to one another, the cervical and lumbar cross-sections are much bigger than the other two, and that is explained due to the cervical and lumbosacral enlargements that cause the cross-sections to be wider. Remember that the enlargements are the regions that supply the limbs, which are the regions that require the most sensory motor processing, thus making them anatomically bigger. Now, to orient ourselves during our next topics, I want to begin by discussing the main anatomical landmarks that the cross-section has, and to do so, it's important that we orient ourselves with the navigational terms. From our last discussion that we had on spinal nerves, you will remember that the orientation we are currently in is from ventral or anterior on this side, and it goes towards the dorsal or posterior side. Additionally, because of the bilateral structure of the spinal cord, the midline point divides the two lateral sides like this. This orientation is important to keep in mind because a lot of the terminology is based on that. Alright, now as a preemptive note, I want to warn you that while covering the internal anatomy of the spinal cord, the figures will get rather dense pretty quickly, so make sure to go slowly if these terms are new for you. With this being said, Starting from the spinal nerve that carries both sensory and motor information of the somatic and autonomic nervous system, you will remember that the spinal cord splits in two structures. One called the dorsal root ganglion that contains the cell bodies of the sensory afferents, and the ventral root that contains the axons of somatic and autonomic efferent neurons leaving the spinal cord. Inside the spinal cord, you will notice that there are two different shades of color, and these represent gray and white matter. To understand the difference between the two, we need to recall the structure of a neuron, which you can see in the bottom right. So, gray matter, as shown by the beige color in the center of the spinal cord, corresponds to the cell bodies of the neurons. Hence, the H or butterfly-shaped structure in the middle is mostly all cell bodies. On the other hand, the white contour that you see is white matter, and it is called that way because the myelin sheets of myelinated axons make it appear clearer in imaging studies. Recall from our discussion about neurons that myelin sheets are produced by two types of glial cells, namely Schwann cells that myelinate neurons in the peripheral nervous system, and oligodendrocytes that myelinate neurons in the central nervous system. As a structure, myelin wraps around the axon and generates these zones called nodes of Ranvier that contribute greatly in increasing the velocity of propagation of the action potential. Two other anatomical landmarks that are important to mention are the posterior median sulcus and the anterior median fissure. Now, with this established, let's cover the divisions of the gray and white matter, starting with the gray matter. The upper dorsal portion of the gray matter is called the dorsal or posterior gray horn, and it corresponds to the terminal zone for the somatic and visceral sensory neurons. Basically, neurons in this region receive the inputs coming from the periphery and then send it up to the brain or brainstem for further processing. The two shades of blue differentiate where the somatic information and visceral information generally go to. As we will see shortly with some more precise divisions of the gray matter, somatic information contacts neurons in the more dorsal part, whereas the visceral inputs go to more ventral parts in the light blue region. The bottom ventral portion of the gray matter is called the ventral or anterior gray horn, and it contains the cell bodies of somatic motor neurons that activate our striated muscles. In between the dorsal and ventral horns, we can find the intermediate zone, and the two most important components that it contains are the cell bodies of preganglionic sympathetic and parasympathetic efferent neurons. Now, the sympathetic cell bodies are only found in segments T1 to L3, and for the parasympathetic system, they are found in segments S2 to S4. In terms of terminology, the sympathetic cell bodies produce an additional bump on the gray matter that is often called the lateral horn, but outside of T1 to L3, it is not found. As a quick shorthand, it is generally safe to reduce the dorsal part to sensory processing and the ventral part to motor processing. These dorsal, ventral, and intermediate zone divisions of the gray matter that I have just introduced can be further divided into 10 more specific sections that are called rex laminae. By the way, as a quick side note, the word rext comes from the last name of the neuroanatomist that came up with these divisions. Now, as you will see shortly, some of the rex divisions contain clusters of neuron cell bodies that share some given property, and as a collective, they are referred to as a nucleus. The terminology to refer to a group of neuron cell bodies as a nucleus is only done in the structures that are a part of the central nervous system. Remember that clusters of neuron cell bodies in the peripheral nervous system were referred to as ganglions. A good example of that was the dorsal root ganglion, which is seen just outside of the spinal cord. Another important aspect about these nuclei to keep in mind is that how they group is not necessarily always in spherical lumps, but it can also be done in columns, and for that reason, some of the nuclei we will encounter are actually called columns, and that is because they span multiple segments in the shape of a column. Finally, I want to let you know that in this video, I will mostly offer an introductory overview for each division without going extensively in the details of each nuclei or lamina, because we will revisit these divisions in their respective contexts, and in these videos about either the somatosensory system or the motor system, it will feel much more relevant to discuss their specifics. Nonetheless, we can start with the rex laminae that are contained in the dorsal horn, which are the first 6. The 6 laminae are somewhat organized in horizontal layers, and as you might suspect, they will play some role in processing sensory information. Rex lamina 1 is the most dorsal lamina, and it contains the posterior marginal nucleus, which receives information about pain and temperature. The second lamina is also known as the substantia gelatinosa of Rolando, which is a nucleus that receives pain, temperature, and touch information. Lamina 3 and 4 can be grouped together as they share the nucleus proprius. This nucleus receives information about pain, temperature, and touch. Lamina 5 and 6 can also be grouped together as they are less precise in their function, but together they allow for processing of touch, proprioception, and pain from visceral organs. You can see more clearly now why the dorsal horn is divided in two different shades of blue. Moving on to lamina 7, it is the only lamina corresponding to the intermediate zone. This lamina has three particular nuclei, namely the interomediolateral nucleus and interomediomedial nucleus that contain the cell bodies of preganglionic sympathetic and parasympathetic efferents. Remember that the sympathetic efferents are from segments T1 to L3 and parasympathetic from S2 to S4. The third nucleus is called Clark's column, and it deals with processing of proprioception. This column is only present from C7 to L3. The next two laminae, lamina 8 and 9, are part of the ventral gray horn and process motor information. Lamina 8 contains motor interneurons that mediate reflex pathways, and lamina 9 contains somatic motor neurons that activate striated muscles. As you can see, there are three general nuclei in lamina 9. The most medial one is responsible for the activation of axial muscles, the central one for proximal limbs, and the most lateral one is responsible for distal muscles. Remember that the regions of the spinal cord that are responsible for our distal limbs are located in the cervical and lumbosacral enlargements, thus the lateral nuclei are only present at these enlargements. Additionally, for all these nuclei, their ventral regions activates extensor muscles and their dorsal region activate their flexor muscles. Again, we will cover all these concepts in much more detail when we will discuss motor systems. Finally, we have the last lamina, lamina 10, that is also known as the gray commissure, and basically the central region is a path that connects the two sides of the spinal cord. Now, it's possible that in your notes or textbooks or whatever resource you use, that you find these sections to be divided differently or described in a different manner in terms of their function, and that is totally normal because although we have a good idea of how the spinal cord operates, there is still a lot to be learned, which makes different sources of information be slightly variable. Accordingly, don't worry if what's written on here doesn't match entirely what you learned. Every rendition of the Rex laminae that I've used to sort of gather and integrate for this video are different to some extent. With this being said, we can move on to the white matter. Recall that white matter corresponds to the axons of the cell bodies of neurons that are in the gray matter. Many axons together can go either up to the brain or brainstem and form what are called ascending tracts, or they can be coming from the brain or brainstem by descending tracts. Essentially, the white matter of the spinal cord acts as an elevator to get information up and down. Much like the gray matter, the white matter is divided into different sections that have their respective functions and processing. Because they are axons and not cell bodies or dendrites, broad white matter sections are referred to as columns, and as you will see later, columns are composed out of tracts. Generally speaking, there are three zones of white matter: the dorsal white column, the lateral white column, and the ventral white columns that each contain multiple tracts. Similar to the Rex laminae, I will offer an overview of these white matter tracts, but note that the more specific details will be discussed in the respective videos. The way we will go about covering them will be to first look at ascending tracts, and then we will go about the descending tracts. Starting with the dorsal column, it only contains ascending tracts, and sometimes just the dorsal column on its own can be considered as an ascending tract. Nonetheless, it is also often divided into the fasciculus gracilis and the fasciculus cuneatus, which altogether carry touch and proprioception information from the body. The fasciculus gracilis is responsible for supplying the lower extremities, so it collects sensory information from T6 and below. For the segments T5 and above, that sensory information is conveyed by the fasciculus cuneatus, which is responsible for upper extremities. You will notice that the fasciculus cuneatus is more lateral than the fasciculus gracilis, and that makes sense because the information about the upper regions is added onto the existing tract for lower limbs. Now, in the lateral column, there are four different ascending tracts: the dorsal and ventral spinocerebellar tracts, the lateral spinothalamic tract, and the spino-olivary tract. In terms of nomenclature, the tracts that have names like this all follow the same rule of the first part being the starting point and the last name being the end point. So, for example, the spinocerebellar tract starts with spino, so the tract begins at the spinal cord and ends in the cerebellum, which is represented by the cerebellar. Similarly, the spinothalamic tract begins in the spinal cord and ends in the thalamus. With respect to their functions, the dorsal division of the spinocerebellar tract is responsible for proprioception for segments C8 to L2, and the ventral part for proprioception for segments L3 to the coccyx. The lateral spinothalamic tract carries mainly pain and temperature information to the thalamus, and lastly, the spino-olivary tract goes from the spinal cord to the inferior olive and is responsible for carrying proprioceptive information. In the ventral column, there is one ascending tract, and it is the ventral or anterior division of the spinothalamic tract. This division carries sensory information about crude touch and pressure. Now, when it comes to the descending tracts, they can be broadly separated into pyramidal and extrapyramidal tracts. The difference in name comes from a structure in the brainstem called the pyramids, which convey the pyramidal tracts, whereas the extrapyramidal tracts take other routes to reach the spinal cord. In the lateral white column, there are three descending pathways: the lateral corticospinal tract, the rubrospinal tract, and the medullary division of the reticulospinal tract. The lateral corticospinal tract is the major tract that descends from the motor regions in the brain to coordinate voluntary fine movements of distal muscles that are associated with our limbs. The rubrospinal tract starts at the red nucleus and descends in the spinal cord to facilitate and modulate the activation of voluntary movements. It mostly does so by activating flexor muscles. The reticulospinal tract has an important role in influencing motor control, posture, and locomotion. It has two divisions, and the one in the lateral column is called the medullary division, which, as you might suspect, arises from the medulla. In the ventral column, there are four descending tracts. The first is the pontine division of the reticulospinal tract that comes from the pons and participates as well in the modulation of movement. Secondly, there is the anterior corticospinal tract that helps coordinating the movements of axial muscles. In third, there is the tectospinal tract that activates muscles of the head and neck to coordinate them with movements of the eyes. Finally, there is the vestibulospinal tract that activates extensor muscles from the signals of the vestibular system to control balance and posture. The last two white matter pathways that are important to point out are the anterior white commissure, which, just like the gray commissure, allows to connect the two halves of the spinal cord together, and that allows for information to cross over. The second pathway is the Lee-Sowers tract, which is known as the dorsolateral fasciculus. Simply put, before the sensory afferents for pain and temperature enter the spinal cord through the spinal nerve, the Lee-Sowers tract allows for these afferents to ascend or descend 1 to 2 vertebral segments before they enter the spinal cord. For example, information that is picked up by the spinal nerve T6 could enter in segments T4 or T8. When we consider all the respective lamina and columns of the white and gray matter, we get a spinal cord that looks something like this. Keep in mind again that this picture is generalized of the actual functional segmentation of the spinal cord, but it nonetheless provides us with a good overview of what's going on there. A final detail that I want to point out is that if we consider back the cross sections from each segment we had in the beginning, you will notice that starting at the sacral segment, as you go up towards the cervical segments, the proportion of white matter increases and the gray matter decreases. Inversely, as you go down, the quantity of white matter decreases and gray matter increases. The simple reasoning for why this occurs can be explained from the fact that as you go up through the spinal cord, the ascending tracts accumulate, and as you are up in the higher levels, the descending tracts have yet to reach their targets, hence causing the gray to white matter ratios to be like this. Inversely, in the more caudal levels of the spinal cord, the ascending tracts have yet to form and the descending tracts have reached most of their targets. With the white matter tracts now covered, we have gone through all the main points that there are to discuss with respect to the spinal cord. The topics we have covered are the external anatomy of the vertebral column, the spinal nerve segments and the associated dermatome map, the anatomy of somatic nerves, the anatomy of sympathetic nerves, and then we went into the internal anatomy of the spinal cord by discussing the general landmarks, the rexed laminae of gray matter, and the white matter tracts. Back to the schematic of the central nervous system structures, we are now ready to cover the next region of the CNS, which is the brainstem. Recall from our discussion about the CNS structures that the brainstem corresponds to the association between the midbrain, the pons, and the medulla oblongata. The brainstem is continuous with the spinal cord and the diencephalon, which, as you might remember, was composed out of the thalamus and the hypothalamus. Sometimes, parts of the diencephalon, such as the thalamus, are included in the brainstem, but for our purposes, we will exclude it and only cover anything that has to do with the diencephalon as part of the forebrain. Now, before we dive into the anatomy of the brainstem, we need to discuss what general roles and functions the brainstem mediates to get a general idea of the structure we will deal with. If you recall our structural and functional model of the nervous system, you will remember that in the most reductive form, the central nervous system, which contains the spinal cord, the brainstem, and the brain, are the intermediate between the perception of our senses and our motor actions. In our discussion about the spinal cord, we discussed at length how spinal nerves that leave the spinal cord carry afferent sensory information and efferent motor information. Additionally, we mentioned how the spinal cord has sensory motor reflexes and central pattern generator circuits that allow to do some processing of the environment. Nonetheless, much of the sensory information that the spinal cord received from the body left the spinal cord through ascending tracts that reached upstream structures like the brainstem and the brain. When it came to motor information coming from the descending tracts, these tracts originated in structures coming from the brainstem or the brain. For that reason, we can say that one of the functions of the brainstem is being a sensory motor intermediate between the brain and the spinal cord, as it contains pretty much every sensory or motor tract that the body uses. Across these tracts, some originate in the brainstem, some get modulated by the brainstem, and some send information to the brainstem for processing. So, although I call the brainstem an intermediate, a lot of processing happens there beyond simply relaying messages throughout the CNS. In addition to this intermediate role, the brainstem also directly receives sensory and sends motor information through structures called cranial nerves. As you can see on the schematic on the left, which is an interior view of the brainstem that we will cover shortly in more detail, cranial nerves are these yellow branches coming out of the brainstem, and they are somewhat similar to spinal nerves because they carry sensory motor information, but instead of supplying our limbs and trunk, cranial nerves supply the face and head region as well as our visceral organs. As a result, there are much more variety in the information they carry. There are 12 pairs of cranial nerves, and 10 of them come directly in and out of the brainstem. Accordingly, the brainstem is often regarded as being the key hub for these cranial nerves and associated nuclei. In terms of more miscellaneous processing, the brain has other nuclei scattered across the medulla, pons, and midbrain that deal with the processing of a wide array of information. To name a few, it contains nuclei responsible for modulating cardiovascular and respiratory activity, pain sensitivity, and other more abstract concepts like alertness, awareness, and consciousness. As you can see, the brainstem is a much more complicated structure in comparison to the spinal cord, as its structure is much less stereotypically organized, and due to its large amounts of different functional units that interact with the spinal cord and the brain, it mediates a lot of roles that are important for our survival. With this introductory overview of the brainstem functions, let's now focus on its external anatomy. To understand where the following diagrams come from, it will be important to recall the neuroanatomical navigation axes. The axis that will be important for us is the posterior dorsal and anterior ventral axis. So, if we first look at the brainstem from this perspective, we get the anterior view of the brainstem, and from this perspective, we get the posterior view. As you might see already, the external surface of the brainstem is much more different from the spinal cord, as it has many more bumps and ridges that will be important to consider. Another axis that will be important for us to keep in mind with the brainstem is the rostral and caudal axis. Remember that rostral is towards the nose and caudal is towards the tail. Accordingly, if we start from the most caudal end of the brainstem, we will get the junction between the spinal cord and the medulla oblongata. Moving rostrally, we obtain the pons, and more rostrally, the midbrain until we reach the diencephalon. Remember that in this video, we won't consider the diencephalon as a structure from the brainstem, and we will cover the details of this division like the thalamus and hypothalamus during our conversation about the brain. Now, in these two figures, there are some important anatomical landmarks that should be pointed out that will be helpful for reference later. First of all, you will notice on the caudal ends of both the anterior and posterior views, the ventral and dorsal roots of the spinal nerves for segments C1 and C2, which mark the most rostral end of the spinal cord. Then, if we consider the interior view of the medulla, most medially we can observe the anterior median fissure that divides the pyramids of the medulla. As we will see later and in other videos regarding the motor system, the medullary pyramids carry descending motor information. In the pyramids, the region where the motor tracts cross is called the pyramidal decussation and represents an important landmark as it generally defines where the medulla and spinal cord divide. Moving laterally from the anterior median fissure, we have the anterolateral sulcus. This division separates the pyramids from the olives, which contain the olivary nuclei, an important cluster of neurons that we will cover later. At the most rostral end of the medulla, there is the medullopontine sulcus that divides the medulla from the pons. Moving rostrally in the brainstem, another prominent structure to note are the cerebral peduncles that are most notably known for carrying descending motor information. In the most medial region of the midbrain diencephalon region, there are two important structures that are characteristic of the interior view: the mammillary body and the pituitary gland that is connected to the hypothalamus by the infundibulum or pituitary stalk. These two structures, along with the thalamus that you can see more rostrally, are part of the diencephalon, so we will see in more detail their roles later. Now, when it comes to the posterior view, in the medulla, it is important to point out the posterior median sulcus that medially separates the two sides. More rostrally in the pons, an important structure is the cerebellar peduncles that connect the brainstem, brain, and spinal cord with the cerebellum. More rostrally in the midbrain, two of the most characteristic structures of the posterior view are the inferior and superior colliculi, which respectively help with the processing of auditory and visual information. Then, rostral to the superior colliculi, we have the pineal body, which is an important structure of the hypothalamus. Now, although for all of these structures that I've pointed out, I will cover their roles later or in other videos, it's good to see their overall shapes and where they are now, because they will act as good reference points when we will consider cross-sections of the brainstem. Finally, the last and maybe one of the most important anatomical detail to consider with respect to the brainstem are the multiple cranial nerves in yellow you see coming out of it, which we are now ready to consider in more detail. If you remember our discussion on the different roles and functions of the brainstem, you will recall that there are 12 pairs of cranial nerves and 10 of them come directly from the brainstem. Now, two important things you will notice with these diagrams is that first, the interior view on the left is much more informative about the cranial nerves in comparison to the posterior view. For that reason, while discussing the cranial nerves, we will mostly consider the interior view. The second thing that you will notice is that from these two diagrams, we can only see 11 out of the 12 cranial nerve pairs. To see the full 12 pairs, we need to consider this ventral or bottom view of the interior surface of the brain. When we compare this figure with the interior view we have been using, I hope that you can see the brainstem in more context and how it's structured with respect to the brain. So, as I said, 10 of the 12 pairs are related to the brainstem, which leaves 2 pairs out of it. These 2 pairs correspond to cranial nerve 1, the olfactory nerve, and cranial nerve 2, the optic nerve. By the way, before we get into it, I want to mention that the 12 pairs are numbered 1 to 12 based on a rostral to caudal direction, meaning that the first cranial nerve will come from the brain and the 12th one will be at the most caudal end of the medulla, so when we will go through them, we will roughly go from a rostral to caudal direction. Also, I want to mention that for any cranial nerve, it is widely accepted to either call them directly by their names or by their cranial nerve number, which is often noted in Roman numeral form, but I personally find it easier to remember them with regular numbers. Finally, you will often see the abbreviation CN, like here for CN1 and CN2, and it simply means cranial nerve 1 and 2. Alright, so back to the olfactory and optic nerves, the two convey purely sensory information to regions out of the brainstem. Starting with the olfactory nerve, we can best appreciate its function through this mid-sagittal diagram of the human face. To get oriented, Here is the tongue and hard palate that illustrate the position of the mouth. Superior to the hard palate, we have the nasal cavity where the olfactory nerve plays an important role to mediate our perception of smell. Indeed, the nasal cavity contains the olfactory epithelium, which contains the cell bodies of olfactory receptor neurons that transduce odorant chemicals into electrical signals. These electrical signals then pass through the cribriform plate via the many olfactory nerves to reach the olfactory bulb. After some processing in the bulb, the signals are sent to different areas of the brain through the olfactory tract. In all, the function of olfactory nerves can pretty much be summarized to them being the carriers of afferent information about olfaction from the olfactory epithelium to the brain. Now, when it comes to the optic nerve, it carries afferent information about vision from the retina to the brain. If you are not familiar with the retina is a layer of cells at the back of your eye, and it contains photoreceptors as well as supporting cells that altogether convert visual information to neural signals. These signals are then funneled into the optic nerve, which connects the eye to the rest of the brain. Obviously, the broader context behind these cranial nerves and some additional information about these diagrams will be explained in more detail in their respective videos when we will cover sensory systems. But for now, this should suffice us for us to understand their roles. Very simply, the olfactory nerves carry sensory information for the sense of smell, and the optic nerve carries sensory information for the sense of vision. So, with these first two cranial nerves covered, we can return and focus on the 10 remaining cranial nerves that come out of the brainstem. But before we go through them one by one to get a general feel for their roles, I want to first show you an overview of all these cranial nerves to point out some aspects that will be important to keep in mind throughout our discussion. The way I have decided to organize this overview is to separate the cranial nerves based on the region they leave from in the interior view. Accordingly, the first region is outside of the brainstem, which contains the first two cranial nerves we have covered. Then we move into the midbrain where cranial nerves 3 and 4 exit. Technically, cranial nerve 3 exits at the midbrain Pons junction, but to say it leaves at the midbrain is good enough for us. In the Pons, the only cranial nerve to exit at this level is cranial nerve 5, and at the pontine medulla junction, there are 3 nerves from cranial nerve 6 to 8 that leave. Finally, the 4 remaining nerves exit from the medulla. The first important aspect that I want you to notice is the fact that cranial nerves can either carry sensory information like the first two we have seen, but they can also carry motor information, or in some instances, they can be mixed and carry both. This directly contrasts what we have seen with spinal nerves, as they were all mixed nerves that carry both sensory and motor information. Now, although some nerves are very specialized for one specific task, like cranial nerves 1, 2, 4, 6, 8, and 12, some mediate a much more diverse set of functions, especially 7, 9, and 10. Accordingly, a given cranial nerve can contain multiple different fibers that go on to operate their respective functions. Depending on where these fibers go or come from, they can be categorized in 7 different modalities that describe what type of information is being transmitted. The 7 different modalities are represented by the 3-letter abbreviations you see before each function. To understand what these abbreviations mean, recall that, generally speaking, the fibers entering the central nervous system, or in this case the brainstem, are called afferent fibers and carry sensory information. On the other hand, the fibers that emerge from the brainstem are called efferent fibers and carry motor or autonomic information. Both the afferent and efferent fibers can then further be divided into 7 different types of fibers, with 4 being sensory and 3 being motor. Now, you will notice that for all of these 7 fiber types, the first word will either be general or special, and the second word will either be somatic or visceral. If we start with the second word first, the difference between somatic and visceral is that somatic relates to the body and more precisely to the skin, muscles, and joints, whereas the visceral relates to the internal organs. As a result, all the fibers that go, let's say, to the heart or the lungs will be classified as visceral because they go to internal organs, but any fibers that goes to the skin will be labeled as somatic. For general and special fibers, let's start with sensory fibers first. In this instance, general afferent fibers are the ones that carry information about touch, pain, temperature, and proprioception, which primarily comes from the skin, muscles, and joints, whereas special fibers carry information from our special senses like vision, hearing, balance, olfaction, and taste. In terms of efferent fibers, General efferent fibers send information to skeletal, smooth, and cardiac muscles, as well as glands. Special efferent fibers, on the other hand, activate muscles that are derived from pharyngeal arches, which are basically the muscles that constitute the face, the pharynx, the larynx, and a few other structures in the head. Altogether, the main difference between general and special fibers is whether or not spinal nerves are also capable of carrying the information. For example, the senses coming from our head like vision, olfaction, and so on are only carried by cranial nerves, but our senses of touch, pain, and proprioception are both carried by cranial and spinal nerves, which is why only the fibers carrying vision and so on are said to be special, and the same logic goes for efferent fibers. In summary, all 7 distinctions of fibers can be summarized by these 3 pairs of words, with the first being either special or general, the second being either somatic or visceral, and the third being either afferent or efferent. Accordingly, as long as you know the difference between each word and what regions they correspond to, it should be easier to derive the different types of fibers. The only difference I would like to explicitly point out is between special visceral afferents and special somatic afferents. As you can see, SVA fibers in green are responsible for olfaction and taste, whereas SSA fibers in blue are responsible for vision, audition, and balance. The key difference between them is the somatic and visceral distinction, and that is explained by the fact that the senses supplied by the SSA fibers are derived from ectoderm, whereas the senses supplied by SVA fibers are derived from endoderm. With the exception of this distinction between these 2 types of fibers, the rest should be straightforward to determine. Now, although I have decided to organize the cranial nerves based on where they exit in the brainstem, it can also be good practice to reorganize the cranial nerves based on their modalities to understand how they compare to one another. This can for sure help study their functions and maybe highlight commonalities or differences that are not apparent under this view. The final aspect that I want to discuss before we jump into specifics of the cranial nerves concerns what actually happens inside the brainstem. Indeed, although the external surface of the interior view we have been using shows us the nerves that leave the brainstem, it is also important for us to consider the nuclei that the nerves target or originate from. To do so, we will consider the posterior view, in which we can see the many nuclei that fill the brainstem. Remember that a nucleus is defined in the central nervous system as a cluster of neuronal cell bodies that usually share some kind of feature. The 2 broad distinctions for the nuclei in the brainstem are sensory and motor. In this figure, the sensory nuclei are located on the left and the motor nuclei are located on the right. All of these nuclei are obviously bilateral but are shown on one side for simplicity. Broadly speaking, sensory nuclei are the targets for afferent cranial nerves, and motor nuclei hold the cell bodies of the neurons that constitute efferent cranial nerves, but it happens in some instances where the nuclei are somewhat mixed. In terms of their disposition, sensory nuclei are generally found laterally, whereas motor nuclei tend to be more medial. An important aspect between cranial nerve and nuclei, not to be confused about, is that there is not a one-to-one correspondence between nerves and nuclei. For example, the trochlear nucleus entirely supplies the trochlear nerve, but in other instances, there are nuclei such as the nucleus solitarius that integrates information from many cranial nerves. Accordingly, it will be very important to take the different nuclei of the cranial nerves into account to get an appreciation of their function. With this overview now established, we can begin our discussion about the cranial nerves in the brainstem, starting with cranial nerve 3, the ocular motor nerve, cranial nerve 4, the trochlear nerve, and cranial nerve 6, the abducens nerve. These 3 nerves generally fall under the umbrella of ocular motor nerves, as they are responsible for the movements of the eye. To discuss these 3 cranial nerves, let's first take 3 cross-sections of the brainstem to see where their nuclei reside, because it turns out that each nerve has its respective nucleus. Starting with the ocular motor nerve, we can find the ocular motor nucleus at the level of the superior colliculus in the midbrain. A little more caudally at the level of the inferior colliculus, we can find the trochlear nucleus, and finally we can find the abducens nucleus. Since all of these nuclei supply motor nerves only, they house the cell bodies of the neurons that will send projections that will exit the brainstem. One particularity you will notice with the projections coming from the trochlear nucleus is that first, they cross and they go to the opposite side from where they originate from, while the projections from the oculomotor and abducens nucleus stay on the same side. This means that the projections from the left trochlear nucleus will reach the right eye, and the projections from the right trochlear nucleus will reach the left eye. Secondly, the projections from the trochlear nucleus leave the brainstem from the posterior side. This aspect is very characteristic of the trochlear nerve, as it is the only cranial nerve leaving from the posterior side. Now, in terms of their function, as I said, they are responsible for the movements of the eye, so let's consider the muscles that generate the movements. It turns out that all of the possible motions of our eyes are mediated by 6 different muscles: the superior, medial, inferior, and lateral recti, as well as the superior and inferior obliques. The superior oblique is a bit different from the other muscles, as it contracts the eye through a small tendon called the trochlea. When we consider what cranial nerves innervate what muscles, cranial nerve 4 and cranial nerve 6 are each responsible for one of the 6 eye muscles. As you might guess, due to the trochlear nerve innervates the superior oblique. On the other hand, the abducens nerve innervates the lateral rectus, and the 4 remaining muscles are innervated by cranial nerve 3, the oculomotor nerve. Since all these muscles are striated, the fibers that innervate the muscles are labeled as general somatic efferent, or simply GSE. Now, as far as this video goes, we won't discuss the process of eye movement, but note that all 3 cranial nerves interact with each other in some antagonistic way to produce fluid eye movements. To do so, the 3 nuclei communicate with each other to coordinate the movements, and this is done through the medial longitudinal fasciculus, which is a white matter tract that integrates their signals with other brain centers to mediate fluid eye movements. Beyond controlling the muscles that move the eye, the oculomotor nerve has other functions that are important to consider, which we can observe best with a lateral view of the eye. You will recognize with the orange and blue colors the respective muscles each cranial nerve innervates. Along with the muscles, the lateral view also includes the oculomotor nerve, which divides into a superior and inferior branch when it reaches the region of the eye. The inferior branch innervates the inferior rectus, the inferior oblique, and the medial rectus. The superior branch innervates the superior rectus and the levator palpebrae superioris. The levator palpebrae superioris is a muscle outside of the 6 that control eye movements, and it is responsible for raising the eyelid when it is contracted. To understand the final function of the oculomotor nerve, it is important to understand that the fibers that travel inside of the oculomotor nerve do not only originate from the oculomotor nucleus, as it turns out that the oculomotor nerve also carries fibers that originate in the Edinger-Westphal nucleus. The Edinger-Westphal nucleus gives rise to the preganglionic parasympathetic fibers that travel through the oculomotor nerve to reach a structure called the ciliary ganglion, which lies close to the optic nerve, or as we've seen, cranial nerve 2. By the way, remember that in the peripheral nervous system, ganglions are defined as a collection of neuronal cell bodies. Now, if we take a closer look at the eye, the ciliary ganglion contains postsynaptic fibers that, when activated by the presynaptic fibers of the Edinger-Westphal nucleus, will activate the ciliary muscle to constrict the pupil. The constriction of the pupil will have the effect of reducing the amount of light entering the eye, and this is an important effect of the parasympathetic response. Additionally, the contraction of the ciliary muscle can change the shape of the lens, and that allows for the ability of our eyes to focus light from objects at varying distance on our retina. This mechanism is also known as accommodation. In all, this packed diagram should summarize the main functions and ideas behind cranial nerves 3, 4, and 6. With these 3 cranial nerves covered, we can move on to the next cranial nerve, the trigeminal nerve. In comparison to the other cranial nerves, the trigeminal nerve is very special because its respective nucleus is located all over the brainstem, as you can see in green. In total, there are 4 divisions of the trigeminal nucleus, and to see them more clearly, let's take a cross-section at the level of the midbrain, pons, and medulla. Starting at the pons, we can find 2 divisions, namely the principal sensory nucleus in orange and the motor nucleus in red. In terms of their functions, the principal sensory nucleus mostly receives sensory information about touch from the face and proprioception from the jaw. On the other hand, The motor nucleus is responsible for innervating the muscles of the first pharyngeal arch, which are mainly the muscles of mastication, and the tensor tympani, a small muscle in the ear. The motor nucleus also will send projections to innervate other muscles such as the tensor villi palatini, the mylohyoid, and the anterior belly of the digastric, but the mastication muscles and the tensor tympani are the most relevant for our purposes. If we go at the medulla level, we can find the spinal nucleus, which is an important sensory nucleus that receives information about touch, pain, temperature, and proprioception for the entire face. Finally, at the level of the midbrain, we can find the mesencephalic nucleus that processes mainly proprioceptive signals from mastication, neck, and head muscles. In all, out of the 4 nuclei, 3 are sensory and 1 is motor. Now, an important characteristic of the trigeminal nerve is that it turns out that it is actually the largest cranial nerve out of them all. This is in part caused by the fact that the sensory signals coming from the face that innervate the trigeminal nuclei come from the trigeminal ganglion, which is best appreciated on the lateral surface of the face. The trigeminal ganglion is to some extent analogous to the dorsal root ganglion that we have covered in our discussion about the spinal cord, as it contains the cell bodies of the pseudo-unipolar neurons that will reach each sensory nuclei. You will notice that the trigeminal ganglion has 3 different branches and each innervate a specific region. If you remember again from our discussion on spinal nerves, these specific regions are called dermatomes and essentially correspond to the regions of the body that are supplied by specific divisions of nerves. The first and most superior branch of the trigeminal ganglion is called the ophthalmic nerve and supplies the upper portion of the head. As a shorthand, this division is abbreviated to V1. As you might imagine, the next division is labeled V2 and it corresponds to the maxillary nerve. Finally, the last division, V3, is the mandibular nerve. With these 3 dermatomes, we can complete our picture about the dermatomes for the entire body when we consider the dermatomes from the cervical segments that innervate the neck and the back of the head. As a final note, I want to mention that the projections from the motor nucleus travel exclusively through the mandibular nerve division, or V3, to reach their targets. This last detail completes our picture about the trigeminal nerve, which means we are ready to move on to the next cranial nerve, the facial nerve. To cover its functions, let's take 2 cross-sections, one at the level of the pons and one at the level of the medulla. In the pons, we can first find the motor nucleus of the facial nerve, which sends projections to innervate the muscles of the second pharyngeal arch, and these correspond mostly to the muscles responsible for facial expressions and the stapedius, a small muscle in the ear. There are other muscles in the second pharyngeal arch, such as the posterior belly of digastric and the stylohyoid, but for our purposes, the muscles of facial expressions and the stapedius are the most relevant. In the pons again, there is a second nucleus that produces fibers for the facial nerve. It is called the superior salivatory nucleus. The superior salivatory nucleus is responsible for sending preganglionic parasympathetic fibers to 2 ganglions that then relay the signal on important glands of the face to make them secrete their respective fluids. The first ganglion is called the pterygopalatine ganglion, and it connects the lacrimal and mucosal glands of the oral and nasal cavities. The lacrimal glands are located near the eyes and produce lacrimal fluid, more commonly known as tears. The mucosal glands can be found in the sinuses of the face and produce mucus. The second ganglion that is connected to the superior salivatory nucleus is the submandibular ganglion, and its activation activates the submandibular and sublingual salivary glands. As you might suspect, these glands produce saliva. I should also mention that there is a third salivary gland called the parotid gland, but this one gets activated by cranial nerve 9. Now, when it comes to the medulla, it contains an important sensory nuclei for the facial nerve, namely the nucleus solitarius. This nucleus receives taste sensations of the anterior 2/3 of the tongue from the facial nerve. The final detail that I want to mention with respect to the facial nerve is that it also conducts sensory fibers. If you recall the spinal nucleus of the trigeminal nerve, you will remember that its main function was to be a receptive zone to the touch, pain, temperature, and proprioceptive sensations of the face. It turns out that the facial nerve feeds this receptive zone by carrying GSA fibers that innervate small structures in the ear, such as the tympanic membrane, the external auditory meatus, and a small portion of the auricle. Although this detail might not be the most memorable feature of the facial nerve, it is good to keep in mind that cranial nerve 7 also supplies some sensory sensations in the ear. So, with this detail now covered, we have went through most of the essentials of the facial nerve. We can now shift our attention on cranial nerve 8, the vestibulocochlear nerve. So, in comparison to the other cranial nerves found in the brainstem, the vestibulocochlear nerve is rather simple because it is primarily sensory and it innervates mostly one structure, the inner ear. To find that inner ear, we need to first consider the general anatomy of the ear. The ear is generally divided into 3 sections: the outer ear, the middle ear, and the inner ear. Broadly speaking, the general function of the outer and middle ear is to funnel sounds on the inner ear. Now, the inner ear itself is divided into 2 principal functional units. First, there is the snail-shaped structure called the cochlea, which, when we consider its cross-section, contains specialized cells that transduce the vibrations from sounds into neural signals. These neural signals are then picked up by the cochlear division of the vestibulocochlear nerve and carried to the central nervous system to create our auditory perception. The second functional unit of the inner ear is the vestibular system, which is generally composed out of the semicircular canals and the otolith organs. These structures contain specialized cells as well, but do not process sounds. Rather, the specialized cells in the vestibular system sense our motion to produce our sense of balance. The signals picked up from the vestibular system in the inner ear are carried to the CNS via the vestibular division of cranial nerve 8. Altogether, the cochlear and vestibular divisions travel together to reach the pontine medullar junction to synapse on their respective nuclei. Of course, there is much more information to be said about the anatomy and physiology of the ear, which we will do in due time when we will cover the auditory and vestibular systems. One detail, however, that I wish to mention before we move on concerns the 2 muscles you might see in the middle ear, the stapedius and the tensor tympani. If you recall our discussion about the trigeminal and facial nerves, you might remember these names as they were the muscles that the respective motor nuclei from cranial nerve 5 and 7 innervated. With respect to their function, these muscles are very important because they contact tiny bones in the middle ear that conduct the sound. So, when these muscles contract, they dampen the intensity with which the sounds are transmitted to the inner ear. As you can imagine, any deficit with these muscles can lead to very severe conditions because the sounds will be perceived as much louder and might cause great amount of pain. Alright, now the next cranial nerve we will go over is the glossopharyngeal nerve, which, as you can see, has many different fibers that mediate a bunch of different roles in the body. Accordingly, the glossopharyngeal nerve is supplied by many nuclei in the brainstem. Fortunately for us, we can all find them at roughly the same cross-section in the rostral medulla. The first important nucleus that provides fibers to the glossopharyngeal nerve is the nucleus ambiguus. This nucleus provides the origin for the motor neurons that will innervate the stylopharyngeus, which is the only muscle part of the muscles derived from the third pharyngeal arch. When the stylopharyngeus contracts, it allows to elevate the pharynx and the larynx, which in return facilitates the process of swallowing. The second nucleus that provides fibers to the glossopharyngeal nerve is the inferior salivatory nucleus. This nucleus provides preganglionic parasympathetic fibers to the optic ganglion, which then sends postganglionic fibers onto the parotid gland. Recall from our discussion on the facial nerve that the parotid gland was the third salivary gland and that the 2 other glands, the sublingual and submandibular glands, were activated by the facial nerve. The third important nucleus is the nucleus solitarius, which primarily receives sensory inputs from the glossopharyngeal nerves. Some of these fibers are visceral afferent fibers and bring taste sensations of the posterior 1/3 of the tongue. Remember that the remaining taste sensations from the anterior 2/3 were carried by the facial nerve. In addition to these fibers, the glossopharyngeal nerve also carries general visceral afferents that innervate the carotid body and the carotid sinus. To find these 2 structures, we can consider how the heart provides oxygenated blood to the head and the neck. The main blood supply originates in the common carotid artery and then branches into the internal and external carotid arteries. At the bifurcation of the internal and external carotid arteries is where both the carotid sinus and body are located. Starting with the carotid sinus, it is a dilation at the base of the internal carotid artery that contains specialized cells called baroreceptors, which have the primary function of sensing blood pressure. Still at the bifurcation, we can also find the carotid body. The structure is essentially a cluster of specialized cells called peripheral chemoreceptors. These peripheral chemoreceptors have the important role of sensing the blood's partial pressure of oxygen and carbon dioxide, as well as the blood's pH. The final important nucleus of the glossopharyngeal nerve is the spinal nucleus of the trigeminal nerve. Again, recall that this nucleus receives all kinds of somatic afferent information such as touch, pain, temperature, and proprioception from the face. It turns out that the glossopharyngeal nerve also feeds information into this nucleus by supplying somatic information about some structures in the ear, tongue, and upper pharynx. Given that the pharynx is considered to be part of the viscera, these specific fibers going to innervate the structures of the upper pharynx are considered to be general visceral afferent. This final detail completes our general picture of the glossopharyngeal nerve. With the glossopharyngeal nerve now covered, we can move on to cranial nerve 10, the vagus nerve. The vagus nerve is in some way a bit similar to the glossopharyngeal nerve, as all of the nuclei from where the different fibers originate or terminate can be found in the same cross-section in the rostral medulla. Also, much like the glossopharyngeal nerve, the vagus nerve is a mixed nerve carrying both afferent and efferent signals, but the vagus nerve differs from others as it is the only one that leaves the head and neck region of the body. As a result, it is important to note that the vagus nerve is the longest cranial nerve. This aspect can be appreciated from the first important nuclei, the dorsal nucleus of vagus. This nucleus provides parasympathetic innervation to the gastrointestinal tract, the lungs, and the heart. To be more precise, the more extensive list of structures that it supplies are the following, which you can consider if you need to. Remember that the main purpose of the parasympathetic nervous system is to promote digestion and slow down movement to conserve energy. It turns out that the vagus nerve carries the vast majority of all parasympathetic fibers, and as such, its main function is often associated with these fibers from the dorsal nucleus of vagus. One detail I'd like to point out is that this parasympathetic supply from the vagus nerve stops at the splenic flexure of the large intestine. Now, the second important nucleus is the nucleus ambiguus, and its most notable function through the vagus nerve is providing special visceral efferent fibers that will innervate muscles derived from the 4th and 6th pharyngeal arches. Broadly speaking, there are some exceptions, but the muscles of the 4th and 6th pharyngeal arches are basically the constrictor muscles of the pharynx, muscles of the soft palate, and some intrinsic muscles of the larynx. To get a quick appreciation of what these muscles mediate, we can consider this lateral view where we can see the pharyngeal constrictor muscles being attached to the larynx. When they contract, these muscles allow to constrict the esophagus, and that results in facilitating the process of swallowing, also known as deglutition. As for the intrinsic muscles of the larynx, their contraction contributes to the production of speech, or in other words, phonation. The second important role of the nucleus ambiguus is to send preganglionic parasympathetic fibers to the heart, and the downstream effect of that will be to decrease the heart rate. The next nucleus that has important implications for the vagus nerve is the nucleus solitarius, which receives many different sensory signals from the vagus nerve. The first important sensory signal comes from the taste receptors located in the epiglottis and pharyngeal region that transmit taste. Recall that taste from the tongue was carried out to the nucleus solitarius via cranial nerves 7 and 9. The second sensory signal that the vagus nerve carries to the nucleus solitarius is homeostatic information from the aortic bodies and baroreceptors. These structures in question are located at the aortic arch, which is the portion of the aorta that leaves the heart. Much like the carotid body and sinus, the baroreceptors and chemoreceptors that the vagus nerve innervates respectively signals information about blood pressure and about the partial pressures of oxygen and carbon dioxide in the blood and its pH. In addition to these sensory signals, the nucleus solitarius also receives visceral sensations with the exception of pain from the internal organs. Now, the final nucleus that is important to the vagus nerve is the spinal nucleus of the trigeminal nerve, which here again integrates information about touch, pain, temperature, and proprioception for the entire face. The vagus nerve brings to this nucleus general sensory afferent information from the outer ear, the tympanic membrane, the dura of the posterior cranial fossa, which is the cranial cavity at the base of the skull, and from the mucosa of the larynx. Again, since the larynx is part of the viscera, these sensory fibers are labeled as general visceral afferents. As you can see from this diagram right now, the vagus nerve mediates a ton of functions, and accordingly so, there is a ton of information displayed currently. So, make sure to go through it slowly if it's the first time you see these terms. Alright, we now have the last two cranial nerves, cranial nerve 11 and 12, to cover. Since they are rather simple as they mediate mostly one function, we will cover them together. Starting with the accessory nerve first, it is an interesting nerve with respect to the others because it contains both a spinal and a cranial division. As you might imagine, to find the spinal division, we need to consider a cross-section of the spinal cord to find the spinal accessory nucleus and the lateral horns. This nucleus is essentially a row of motor neurons that span from cervical segment C1 to C5. In contrast to the motor neurons we have seen that exit the spinal cord through the ventral root, the motor neurons in the spinal accessory nucleus exit through the spinal accessory nerve, which lies between the ventral and dorsal roots, to innervate two distinct muscles, the sternocleidomastoid and the trapezius. When it contracts, the sternocleidomastoid muscle has the function of rotating the head and flexing the neck. When the trapezius contracts, its main function is to move the scapula, also known as the shoulder blade. Now, the spinal accessory nucleus obviously ends when the brainstem begins, but it is believed that this nucleus is continuous with the nucleus ambiguus in the medulla, and that gives rise to the cranial division of the accessory nucleus. The cranial division of the accessory nerve, coming from the nucleus ambiguus, runs in parallel with the spinal division for a brief period of time, but then shortly after, it separates to innervate some muscles of the soft palate, pharynx, and larynx with cranial nerves 9 and 10. This grouping of nerves, including the cranial nerve 9, 10, and the cranial division of cranial nerve 11, is also known as the pharyngeal plexus. Now, because the core function of the cranial division is somewhat also attributable to the vagus nerve, the accessory nerve is most memorable for its spinal division that activates the sternocleidomastoid and trapezius muscles. Accordingly, if you see the accessory nerve being called the spinal accessory nerve, it simply means that the person behind has chosen to only consider the function of the spinal division. Now, finally, we can move on to the last and 12th cranial nerve, the hypoglossal nerve. Very simply, the fibers of the hypoglossal nerve originate in the hypoglossal nucleus in the medulla, which can be found at the same level as the nucleus ambiguus. This nucleus gives rise to the motor neurons that go on to innervate the intrinsic and extrinsic muscles of the tongue. The only muscle of the tongue not to be innervated by the hypoglossal nerve is the palatoglossus. This final detail completes our discussion on each cranial nerve. Back to this overview, with the information we have accumulated about these cranial nerves, we can now better appreciate the diversity and function that each nerve mediates. We can also appreciate the fact that, for the most part, the fibers that make up these cranial nerves target or originate from a bunch of nuclei in the brainstem, and that makes the brainstem a very complex structure that integrates an immense array of functions. With this being said, however, there is much more about the brainstem we have not covered in our conversation about cranial nerves. So, before we move on to the brain, I want to take a moment to briefly point out some key structures in the brainstem. Obviously, I will not go in much detail about these structures as, for one, if I did it, the video would never end, but most importantly, we will revisit most of these important structures in their respective context. Now, to see some of these structures, let's begin at the medulla, where we will take 3 cross-sections, one at the caudal end of the medulla, one at the middle level of the medulla, and one at the rostral end of the medulla. To get you situated on the anterior-posterior or ventral-dorsal axis, the side pointing towards the bottom is the ventral or anterior side, and the side pointing up is the dorsal or posterior side. This orientation holds for all of the cross-sections of the brainstem and spinal cord you will see in this video. Now, if you recall our discussion on the spinal cord, you will remember that the 2 key components of any cross-section in the nervous system are the gray and white matter. For the most part, gray matter corresponds to the cell bodies of neurons, and white matter corresponds to the axonal projections that are enwrapped in myelin. In the spinal cord, the gray matter had this butterfly or capital H shape and was located in the middle of the cross-section. We further divided the gray matter into 10 divisions called Rex Laminae that each operated a particular task across the spinal cord. When it comes to the brainstem, instead of talking about Rex Laminae, you have seen throughout our discussion on cranial nerves that the gray matter is organized in different nuclei that are scattered all around the brainstem. In the medulla, The two main different types of nuclei that we can observe are sensory and motor nuclei. For the most part, these are all nuclei that we have seen across the cranial nerves profiles. One aspect of the terminology about the gray matter that I'd like to point out, which you might see sometimes in your textbooks, is the concept of the reticular formation. Simply put, the reticular formation represents the central core of nuclei and gray matter of the brainstem. A bit like the butterfly shape in the spinal cord, but considerably more complex due to the different neuronal circuits it contains. For the white matter, the spinal cord was divided in primarily descending or ascending tracts. As a general rule, ascending tracts gathered information from the periphery and carried sensory information to reach targets located in the brainstem, the thalamus, or the cerebellum. Inversely, descending tracts originated in the brain, brainstem, or cerebellum and carried motor information that would eventually reach the periphery. As a result, when we consider the medulla, we will come across a lot of these ascending and descending tracts that carry information to their respective targets. In addition to them, the medulla also includes mixed tracts such as the inferior cerebellopeduncle and the medial longitudinal fasciculus that carries information in both directions. As I said, we will not go through all of these tracts and nuclei in detail until we encounter them in their context, but feel free to use these diagrams to get an idea of what's in the medulla. Of course, I want to point out that these diagrams are rarely anatomically accurate, as, to be honest, the real cross-sections of the brainstem are very messy and hard to anatomically distinguish, which makes every idealized cross-section diagram like this be different. As such, if this diagram differs from your textbook, it's perfectly normal, as every textbook from which I have integrated this information disagrees in some way with one another. Nonetheless, moving on rostrally to the pons, we will consider two cross-sections, one at the caudal level and one more at the middle level of the pons. Here again, just like in the medulla, the pons contain different sets of gray and white matter. The gray matter nuclei can be broadly divided into sensory and motor nuclei, and the white matter that crosses the pons can be divided into ascending, descending, and mixed tracts. In terms of the structure and how things operate, the pons are to some extent similar to the medulla as they contain the same broad types of divisions. Now, where it gets a bit more interesting is at the level of the midbrain. Here, we will consider two cross-sections, one at the level of the inferior colliculus and one more rostrally at the level of the superior colliculus. Before we get into the structures that compose the midbrain, there is one recurrent anatomical aspect that I want to point out. The midbrain is generally divided into three distinct sections. The most dorsal or posterior section is called the tectum, and it corresponds to the combined regions of the inferior and superior colliculi. The most ventral section is called the cerebral peduncles, and the region in between the tectum and the cerebral peduncles is called the tegmentum. Now, at this level of the brainstem, we can find sensory and motor nuclei, as well as ascending, descending, and mixed tracts, but one interesting type of structure that we can start to observe are nuclei that I will label as mixed nuclei. The reason why I call these nuclei mixed is mostly because their projections mediate a ton of different functions that are kind of hard to classify as mostly sensory or motor. If you recall in one of our discussions about neurotransmitters, where we covered the different catecholamines like dopamine, norepinephrine, serotonin, and so on, you might remember that the origin of the neurons that release specific neurotransmitters were localized in some specific regions in the brainstem. For example, we can first consider the locus coeruleus, which is an important nuclei that contains cell bodies of the neurons that release norepinephrine. As we mentioned in the discussion about norepinephrine, the projections coming out of this nucleus go all over the brain and influence different components of our lives, such as our sleep, levels of wakefulness, arousal, and attention. Additionally, it is also known that the locus coeruleus is involved in pain modulation. Another example of this multi-integrative nuclei is the raphe nuclei, which predominantly contains neurons that release serotonin across the central nervous system and influence our emotions, our hunger, our motor movements, and many more. Some other examples include the substantia nigra, which notably releases dopamine and influences our movements and our perception of reward. Again, these are all structures that for the most part, we will encounter them again in upcoming videos about different sensory and motor systems. With the midbrain covered, we can now get a good overview of the anatomy of the brainstem that we went through. We started our discussion by describing the different functions of the brainstem, and then we covered its external anatomy. Then, we moved on to cover the principles behind the cranial nerves, and then we jumped into their individual profiles from cranial nerve 1 to cranial nerve 12. In these discussions, we mostly focused on the associated nuclei and their respective functions without bothering too much about their courses in the body. Finally, we discussed the gray and white matter divisions in the cross-sections of the medulla, pons, and midbrain. This concludes our discussion on the brainstem, and this means we are ready to move on to the next portion of the central nervous system. Based on the embryological divisions, the next section that we will cover corresponds to the prosencephalon or forebrain here in blue. Throughout this video, it will happen that I'll refer to the forebrain or prosencephalon as simply the brain, so make sure not to get confused by that. Additionally, I want to mention briefly that in this discussion, we will not cover the cerebellum because I plan on doing a segment later that will be entirely dedicated to it when we will discuss motor systems. Now, to set the table for our discussion on the brain, I want to take a quick minute to rehash some concepts that we have gone through. First of all, from the structural and functional model of the central nervous system that I have established at the beginning of our conversation, you will remember that in a few words, the general role of the central nervous system, at least as I described it, is to interpret signals from the external and internal environments and generate commands to interact with the environment or to regulate our body. However, as you might suspect, this definition is extremely oversimplified as the entirety of the central nervous system integrates much more information in between to perform tasks that do not necessarily materialize in front of our eyes. For example, our capacity to store memories, feel complex emotions, our abilities to think, create, and process information without acting upon it are mostly all cognitive processes that happen at pretty much every moment of our lives. So, although it is true that a considerable portion of the central nervous system takes care of receiving sensory signals and sending motor outputs, and both of these components are critical to our cognitive processes, our central nervous system does much more. If we consider the individual functions of the spinal cord and the brainstem, you will remember that the two had a few similarities in the fact that they both conducted sensory and motor tracts through their structures, and that they both were the origin or receptors to bundles of neurons called nerves. Nonetheless, it remained that the brainstem was much more complex than the spinal cord because of all the diversity of nuclei it had, and thus the diversity of function it mediates. Also, the brainstem was much more complex because in addition to relaying signals from the brain, it also sent its own projections to modulate cognitive aspects of the brain and the body. Now, it will be no surprise for you to hear that with respect to the brainstem and especially the spinal cord, the jump in complexity for the brain is pretty substantial. As a result, this level of complexity makes it that there are hundreds of different angles that we could take to discuss the properties of the brain, ranging from the different networks that mediate our consciousness to the individual neurons that make all of this possible. Now, because I plan on covering sensory, motor, and more abstract systems of the brain in their respective videos, I want this video to focus on more general topics to give us a good foundation along with the sections on the spinal cord and brainstem, such that when we're covering, let's say, the auditory or visual systems, there will be a good continuity with what we've covered here. Now, throughout this section, you'll see different cross-sections and perspectives of the surface of the brain, and the basis for all of these drawings comes from a software that models what is called the MNI brain. In essence, the software allows you to navigate around with a 3D brain and interact with it from many different perspectives. As a side note, the abbreviation MNI stands for the Montreal Neurological Institute, which is the place that came up with this model, and they did so by taking the average brain structure from different people. If you want to download this software, I have included a link in the description that offers a version for Linux, macOS, and Windows. Alright, so to get started on the anatomy, I first want to review the neuroanatomical navigation terminology that we have established in the very first sections of our discussion because a lot of the upcoming terminology that you will see is based on these terms. If we consider this view of the brain, given that the brain is a three-dimensional structure, we can analyze it by decomposing it into two-dimensional planes that each take different cross-sections of the brain into consideration. In no particular order, the first plane is the horizontal plane and it divides the brain into a top and bottom half. The second plane is the coronal plane, which divides the brain in a front and back half. And finally, the last plane is the sagittal plane, which divides the brain into a left and right half. Now, all these planes produce their respective cross-sections, but to navigate within them, there is additional terminology that is used. In the forward and backwards axis, the direction that goes towards your face is the anterior direction, and the direction that goes towards the back of your head is the posterior direction. In the up-down axis, the direction towards the top of the head is called either dorsal or superior, and with respect to the brain, these two words are pretty much interchangeable, but some anatomical structure use the dorsal while others use the superior, so I will include them both. Likewise, for the bottom direction, the words associated are ventral and inferior. When it comes to the axis that goes towards the sides of your head, the middle point is often referred to as the midline, and as you go away from it, you go towards the lateral directions. This little review should suffice for us while we cover some structures of the brain. One last detail that I want to quickly mention is that in the upcoming sections of this discussion, and even in previous diagrams, I have mostly used two views of the brain: the view of the lateral surface and the view of the medial surface, which can be obtained by taking a sagittal cut right in the middle of the brain to separate the two hemispheres. This cut is also known as the mid-sagittal cut. Throughout this video, we will obviously cover more perspectives and surfaces of the brain, but I wanted to particularly point these two out because they are the most common views in any textbooks covering neuroanatomy. Now, as I said just previously, we can generally divide the brain based on its anatomical landmarks or functional regions. Our ability to divide the brain based on its anatomy comes from the fact that it has an extensively folded surface that produces distinguishable ridges and grooves that create apparent frontiers between regions. Presumably, the reason for this highly folded surface is to be able to fit our very large surface area of gray matter into our skulls for us to be able to process complex cognitive behaviors. To see these ridges and grooves more clearly, we can take a coronal cross-section. As you can see, the surface of the cortex is filled with folds. If we zoom on one of these folds, the first thing that is most apparent is that there is a beige layer and a white layer. The beige layer corresponds to gray matter, and the white layer corresponds to white matter. If you recall our discussion on the brainstem and spinal cord, you will remember that the gray matter corresponded to the cell bodies of the neurons, and the white matter corresponded to the axons of the neurons that are covered in myelin. Now, one particularity that I will mention right now about the brain is that the gray matter that covers the surface of the brain is also referred to as the cortex. Hence, anytime you hear about the cortex or anything being preceded by cortical, it corresponds to the external layer of gray matter. If we go back to the folds of the brain, an important point of terminology is the fact that the grooves, which are the bottom part of the fold, are called sulci or sulcus in singular, and the ridges are called gyri or gyrus in singular. Although the distribution and location of each sulcus varies in each individual, there are some sulci and gyri that we all share for the most part, and thus constitute good anatomical landmarks to navigate around the brain. More importantly, beyond being specific anatomical landmarks, I want to place a special emphasis on the knowledge of the different sulci, because as you can see in the cross-section, most of the gray matter is hidden from the external surface, and given that the gray matter, as we will cover later, is the region where most of the neuronal integration happens, the sulci are very important to consider from a functional perspective. If we first consider the lateral and medial view of the brain, there are four principal frontiers that are very characteristic of the human brain formation. The four are the central sulcus, the lateral or sylvian fissure, the parieto-occipital sulcus, and the preoccipital notch. As a side note, a sulcus and a fissure pretty much represent the same thing, but a fissure is much deeper than a sulcus. Altogether, these four frontiers divide the cortex into four general lobes. The central sulcus divides the frontal and parietal lobes, and the lateral fissure divides the temporal from the frontal and parietal lobes. The parieto-occipital sulcus and the preoccipital notch separate somewhat arbitrarily the occipital lobe from the parietal and temporal lobes. The four lobes represent the broadest way of dividing the brain in terms of anatomy, but as you can see, within each lobe there are many more sulci and gyri that we can use to our advantage to divide the brain further. Let's start with the lateral view. As a preemptive note, this diagram will get filled pretty quickly with anatomical landmarks, so if it's your first time seeing them, make sure to go slowly through them so that you don't get overwhelmed. One tip of advice I can give you to ease the load of learning these regions is to make sure you are comfortable with the neuroanatomical navigational terms because, as you will see, pretty much all of the gyri and sulci have very logical names that are based on their positions. Starting at the central sulcus, if we go in the anterior direction towards the frontal lobe, we will meet two distinctive sulci, the superior and inferior precentral sulci. Together with the central sulcus, these two sulci form the boundaries to the precentral gyrus. The remaining portion of the frontal lobe can be divided into three general gyri: the superior frontal gyrus, the middle frontal gyrus, and the inferior frontal gyrus. The two sulci that divide these regions are the superior and inferior frontal sulci. Within the inferior frontal gyrus, there are some additional important areas that I would like to point out because they mediate important roles in speech production. These areas are called pars opercularis, pars triangularis, and pars orbitalis. The anterior ascending ramus separates the pars opercularis from pars triangularis, and the horizontal ramus separates the pars triangularis from pars orbitalis. Altogether, these regions generally represent the main areas of the frontal lobe. If we go back to the central sulcus, but now we go in the posterior direction to the parietal lobe, we will come across the post-central sulcus, which forms the boundaries with the central sulcus to the post-central gyrus. In a sort of perpendicular orientation to the post-central sulcus, we can observe the intraparietal sulcus, which divides the remaining parietal lobe into the superior and inferior parietal lobules. Again, due to its functional role, the inferior parietal lobule can be further divided into two important regions. The first, more anterior region is the supramarginal gyrus, and the more posterior region is the angular gyrus. Now, if we consider the temporal lobe, its divisions on the lateral surface are much simpler. A bit like the frontal lobe, it is divided into three gyri, the superior, medial, and inferior temporal gyri, and between them we can find the superior and inferior temporal sulci. By the way, I want to mention that one good trick to find the supramarginal and angular gyri is that to find the supramarginal gyrus, you can follow the course of the lateral fissure, and to find the angular gyrus, you can follow the course of the superior temporal sulcus. When it comes to the occipital lobe, we will leave it as it is. There are some sulci on its lateral surface, but they are not the most important ones. This final detail completes our picture for the lateral surface. As I said, I am well aware that there is a lot of terminology and the diagram is loaded with information, but if you go slowly with the neuroanatomy axis in mind, learning these regions should be much easier. Now, when it comes to the medial surface, there is also some important gyri and sulci to consider. One of the most prominent sulci is called the cingulate sulcus and forms the main boundaries for the cingulate gyrus. Along with another sulcus called the paracentral sulcus, the cingulate sulcus also formed the boundaries for the paracentral lobule, which is a shared region between the frontal and parietal lobes. In the parietal lobe, the cingulate sulcus and the parieto-occipital sulcus form the boundaries for the precuneus gyrus. Just like the precentral sulcus that was interior or in front of the central sulcus, the precuneus gyrus is anterior to the cuneus gyrus, which is located in the occipital lobe. In addition to the cuneus gyrus, the occipital lobe also contains the lingual gyrus, which is separated from the cuneus gyrus by the calcarine sulcus. In essence, these areas are the main gyri and sulci of the cortex that are outlined on the medial surface. There are no obvious and prominent sulci in the temporal lobe that we can clearly see from this view. To complete our picture on the relevant sulci and gyri of the brain, I want to consider the ventral and dorsal surfaces of the brain. As a reminder, the dorsal surface is a view from the top and the ventral surface is a view from the bottom. Again, to get it situated, we can find the frontal, temporal, parietal, and occipital lobes across both of these views. Additionally, we can see the central sulcus on the dorsal surface. One aspect that I have kind of taken for granted for the moment, but that we can better appreciate from these two views, is the fact that the brain is composed out of two cerebral hemispheres, a left and a right one. The separation between the two hemispheres is called the longitudinal fissure, and again, remember that a fissure is just a deeper sulci. Now, for the ventral surface, there are a few sulci and gyri in the temporal lobe that I would like to point out. First, recall the three gyri that we have covered in the lateral surface, the superior, middle, and inferior temporal gyri. These gyri are separated by the superior and inferior temporal sulci. Now, medially to these regions, we can find two other important gyri, the parahippocampal gyrus and the fusiform gyrus. The parahippocampal gyrus is separated from the other gyri by the rhinal and collateral sulci, and the fusiform gyrus is separated from the inferior temporal gyrus by the lateral occipitotemporal sulcus. To better see these structures, we can consider a ventral view with no cerebellum and no brainstem. As you can see, the boundary between the temporal and occipital lobe is a bit vague on the ventral surface, and rightfully so, because there are no evident landmarks to divide them, so in this diagram you can take this temporal-occipital division with a grain of salt. Up until now, we have talked a lot about the landmarks of the brain, but not so much about what actually happens within or around them. It turns out that to process the world around us and ourselves, the brain is divided into distinct functional regions that sometimes process very specialized bits of information, or sometimes integrate information from different areas to create complex cognitive functions. What I want to do now that we have surveyed the different sulci and gyri, is to go over the most notable functional units of the brain and very briefly discuss what they do to get an idea of how the brain is organized. Obviously, our more detailed conversation about these areas will come in their respective contexts. The first type of cortical areas that the brain has are sensory areas. These areas include the somatosensory areas, the auditory areas, the visual areas, and the piriform cortex, which is the principal cortical area to process the sense of smell. For the somatosensory, auditory, and visual areas, there is an important principle by which the brain processes sensory information. That principle is the idea of division of labor and functional hierarchy. The most striking and well-studied example happens in the visual areas, which are all primarily located in the occipital lobe. The cortical areas representing the visual areas can be broadly divided into the striate and extrastriate cortex. The striate cortex represents the first stage of visual processing and processes somewhat crude information from the eye, such as the orientation of visual input. Then, this information is passed down to extrastriate areas, so outside of the striate cortex, to process and integrate more complex aspects of vision, such as movement and more complex shapes. A very similar idea occurs in the auditory areas, where you can first find the primary auditory cortex, and then secondary areas that process more complex auditory information. A well-known example of a downstream auditory area is Wernicke's area, which is primarily responsible for the comprehension of speech. In the somatosensory system, which is responsible for processing our somatic sensations like touch, pain, temperature, and proprioception, it also has primary and secondary areas that process different levels of complexity for a given sense. By the way, note that the primary somatosensory cortex is located in the post-central gyrus. Now, the second type of cortical area that the brain possesses are the motor areas. The motor areas are all pretty much located in the frontal lobe and mediate the planning, control, and execution of voluntary movements. Similarly to the sensory areas, the different motor areas of the brain mediate different components of movements and are hierarchically organized. The primary motor cortex, which is located in the precentral gyrus, is considered to be the main output region for motor commands, but the planning and control of movements happens mostly in nearby regions called the premotor cortex and supplementary motor area. Two interesting areas that are part of the premotor cortex are the frontal eye fields, which contribute to proper eye movements, and Broca's area in the inferior frontal gyrus, which is often considered to be responsible for proper speech production. As a side note, through different dissections and imaging studies, it became well known that Broca's and Wernicke's area were connected together to properly mediate speech. The interesting part about this is that if one of these areas is to be damaged, then speech gets impaired in different ways. Such conditions are called either Broca's aphasia or Wernicke's aphasia, depending on where the damage is. Since Broca's area is a motor area and thus responsible for the motor output, Broca's aphasia produces a speech that is hardly understandable because the motor output is impaired and the person has a hard time saying the words. Nonetheless, although patients with Broca's aphasia have a very impaired fluency, their understanding of what is being said to them remains intact. On the other hand, Wernicke's area mediates speech comprehension, and when it is damaged in cases of Wernicke's aphasia, the patients can speak fluently because their Broca's area is intact, but the meaning or the semantics of what they say is usually nonsensical. In addition to that, patients with Wernicke's aphasia have trouble with understanding what is being said to them. These two neurological disorders exemplify very well the idea that the brain is divided into different functional units that communicate with each other to process different cognitive functions. Now, with the main sensory and motor cortical areas covered, you will notice that much of the cortex still remains undescribed. If you recall the beginning of our discussion on the brain, you might remember that the brain and the central nervous system as a whole does much more than simply perceiving and responding to the environment. In between the perception of the world and the output of our actions, much of the brain area is devoted to performing complex and integrative cognitive functions that do not necessarily materialize in front of our eyes. These areas that perform these integrative functions are called association cortices, and each lobe except the occipital one is mostly made up of that. The most notable example of this is the frontal association cortex. In essence, this region of the brain is one of the most important factors that makes us humans, as it processes our working memory, which is our ability to temporarily keep information in our minds. It processes our emotions and various executive functions, such as our ability for reasoning, attention, planning, decision-making, and many more. In the temporal lobe, the temporal association cortex is primarily known for its ability to mediate object recognition and memory due to its close proximity to the hippocampus. In the parietal lobe, the parietal association cortex integrates from all the senses, and it is known to mediate our awareness of our bodies, many spatial functions, and also our ability to read and write language. Even with these descriptors, note that each association cortex mediates much more. The last functional region that I want to point out in this diagram is the limbic system, which can be best seen on the medial surface. The limbic system is a complex circuit of different structures that mediates our emotions, some forms of learning and memory, our sexual behaviors, and many more. Some notable structures that participate in this circuit include the cortical regions of the limbic lobe, so the cingulate and parahippocampal gyrus, the medial and orbital frontal cortex, the amygdala, the thalamus, and the hypothalamus. In this diagram, the brain can obviously be further subdivided into more precise functional areas, but what we have should suffice for us to get the gist of how the brain is believed to work, which is the idea that it is divided into many functional subunits that process relatively simple to very complex information, and they all communicate in some way with each other to create a unified perception of the world. From this discussion, you might have noticed that in the sensory areas, we have not covered where the sense of taste gets processed, and that is because it is hidden in the external surface view. To find where this area is located, we need to take a look inside of the lateral or sylvian fissure to discover an important cortical region of the brain called the insula. We can also appreciate the structure of the insula from a cross-section. As you can see, the insula represents a substantial amount of gray matter, and it is thus important to discuss. The area of the insula spans somewhat essentially the length of the sylvian fissure, and thus processes many functions. To name a few, the insula is responsible for housing the gustatory cortex. It is also reportedly responsible for our sense of self-awareness, our visceral sensations, and homeostatic control, and our ability to process emotions such as compassion and empathy. Because of its hidden nature, there is an anatomical term called the operculum that represents the cortex overlaying the insular cortex. We can find a piece of operculum in the frontal, parietal, and temporal lobes. In the cross-section, the operculum are shown as the following. With the content that we have covered up until now, we have built ourselves a very good intuition on how the brain is organized, both functionally and structurally, but our intuition only holds from the point of view of the external surface, as we didn't discuss much of what's going on inside the brain. Accordingly, I want to take a moment now to survey the locations and functions of some of the most notable structures that are hidden within the brain for us to get a complete overview of the main functional units. Since these structures are buried inside the cortex, they are also known as subcortical structures. To observe most of these important subcortical structures, I want to consider two views. First, the mid-sagittal view of the brain that we are used to seeing, and secondly, a cross-section of the brain at this anterior level. To situate yourself in the cross-section, imagine we are looking at the brain from this direction and the frontal portion is removed. Now, if we start with the mid-sagittal view, we can orient ourselves by pointing out some structures we are already familiar with, starting at the surface where we will find the cortex. In terms of its function, it's kind of hard to describe what the cortex does because it will always feel reductive, but in essence, the cortex integrates information for us to perceive, feel, and act on the environment that surrounds us. Another area that is kind of hard to functionally summarize is the brainstem, which, as it is implied in its name, is located at the base of the brain. The brainstem has three general divisions: the medulla, the pons, and the midbrain. And altogether, they mediate a bunch of different functions, but mainly, they are involved in the homeostasis or balance of the body, the senses and the motor activity of the face and viscera, and the brainstem also has a wide variety of projections that go throughout the brain to participate in different circuits that influence our cognitive states. For more information about the brainstem, make sure to consult our discussion on it, where I elaborate much further on what happens there. Right next to the brainstem, we can also see the cerebellum, which, as we will cover in its respective video, interacts with the rest of the central nervous system to help mediate and coordinate movements. It is also involved in motor learning and other cognitive abilities such as language and memory. As such, many projections go in and out of the cerebellum to interact with the spinal cord, brainstem, and cortex. Now, for the next two subcortical structures, you might remember their location from when we covered the anatomy of the brainstem. The two structures in question, the thalamus and the hypothalamus, are both part of the diencephalon, which is located rostrally with respect to the midbrain. In terms of their function, the two are very complex, so again, a short description might not make justice to the full extent of their roles. In essence, the hypothalamus is mostly known for its involvement in our hormonal regulation via its connections to the endocrine system, but it also mediates a wide variety of functions including regulating our sleep and the homeostasis of our body. In another discussion, I plan on covering the different divisions that make up the hypothalamus, and it contains many different nuclei that perform many different functions. Likewise, for the thalamus, it is essentially an egg-shaped ball of mostly gray matter that contains more than 50 known nuclei, which, as you might guess, mediate different roles depending on which nuclei we are referring to. The one commonality that most of these nuclei have, and contribute to the main function of the thalamus, is that they all receive some type of input and then communicate information with the cortex. As such, the thalamus is involved in communications across the entire cortex and participates to diverse aspects of cognition. One thing I want to quickly address with respect to the function of the thalamus is that some of its most popular divisions are those that specifically relay certain types of information to the neocortex. For example, it contains a few sensory nuclei that relay, for instance, visual information from the eye to the primary visual cortex, or information from the ears that passes through the brainstem to the primary auditory cortex, and as such, the thalamus has been labeled as a relay station. However, the thalamus is much more complex. Beyond sending information to the sensory areas, it also communicates with association cortices that mediate much more complex functions. These types of signals that come out of the thalamus are called feed-forward because in terms of hierarchy, the neocortex is above the thalamus, and as such, it feeds forwards to the top. In terms of its processing, it has been found that the thalamus acts as a gatekeeper for information, so the feed-forward connections from the thalamus should not be seen as mere relays, but more integrative in nature. Additionally, the thalamus receives an incredible amount of feedback projections from pretty much the majority of the neocortex, and this element too adds an additional level of complexity to the thalamus. The next important structure that I want to point out is the basal ganglia. The basal ganglia is a collection or circuit of nuclei that work together to control movement initiation. This circuit is also involved in some forms of implicit memory, habit learning, as well as other diverse aspects of cognition. Most of the components of the basal ganglia can be best appreciated in the cross-section, where we can find the caudate nucleus, the putamen, and the globus pallidus. In addition to that, the basal ganglia is also composed out of the substantia nigra, which is a particular nucleus in the midbrain that we came across in our discussion on the brainstem. The basal ganglia contains other structures that participate in its circuit, but these should suffice for us to get an idea of where the basal ganglia is located. In this current cross-section, we can also see another important group of nuclei called the amygdala. The amygdala is a structure we have already encountered when we talked about the functional anatomy of the cortex, and it was an important component of the limbic system. Through various lesion and imaging studies, it was discovered that the amygdala is involved in memory, emotions, fear, anxiety, decision-making, and many more functions of that nature. Now, as of yet, our discussion on the different structures of the brain has mostly focused on the gray matter, but the brain has also some important white matter structures that we need to consider. The most prominent and well-known white matter tract in the brain is called the corpus callosum, and it is located here. The main function of the corpus callosum is to provide a pathway for the two hemispheres to exchange information. This can be best observed in the cross-section, where we can see the corpus callosum directly bridge the left and right hemispheres. In the cross-section, there are a few other important white matter pathways that we can point out. First, there is the anterior commissure, which connects the two temporal lobes. Then, we can also find the internal capsule, which is an important bilateral white matter pathway that connects the cortex with the brainstem, spinal cord, and other subcortical structures. On the lateral side of the putamen and globus pallidus, we can also find the external capsule, which carries cortico-cortical association fibers. Here, cortico-cortical simply means that it carries information from one region of the cortex to another region of the cortex. The final subcortical structure that I want to point out is the hippocampus, but to see it, we need to consider a more posterior cross-section at the level of the thalamus. The hippocampus will be located in this medial region, and in terms of its function, it is mostly known for its involvement in diverse aspects of memory. The output of the hippocampus is then carried out by a white matter tract called the fornix, which is located ventral to the corpus callosum. This last bit of information should be enough for us to understand the main functions of the different subcortical structures. Alright, now that we have an idea of how the cortex is divided, let's consider what happens inside the gray matter to make all of this possible. To get us started on how our knowledge of the cortex came about, I want to first consider some of the different staining techniques that have been developed over time to visualize neurons. In the field of neuroscience, two of the most historically notable staining techniques are the Nissl stain and the Weigert stain. The Nissl stain, developed by Franz Nissl, aims to stain RNA by exploiting the fact that RNA is negatively charged. Now, since most of the RNA is processed at the endoplasmic reticulum, and the endoplasmic reticulum is concentrated in the soma or cell body, the Nissl stain ends up staining the cell bodies of the neurons. As a result, if one uses the Nissl stain on the gray matter of the cortex, the result will be the following. As you can see, the cortex is filled with cell bodies of different shapes and sizes. More formally, the study of the cellular properties of the gray matter in the CNS is called cytoarchitecture. The second important staining technique, the Weigert stain, developed by Carl Weigert, stains myelin sheets, and as a result, it allows to visualize white matter projections in the cortex. The study of the different white matter projections is also known as myeloarchitecture. As you can see, each stain allows you to get different information about the properties of the neurons you are studying. Out of these two stains, the Nissl stain holds a particular importance in neuroscience, as this method was used by a neuropsychiatrist named Corbinian Brodmann, who stained different areas of the cortex to categorize them based on the properties of the cell bodies. As a result, the scientist came up with a map called the Brodmann map. Now, although the Brodmann map has been criticized and has some glaring flaws, the terminology that it brought with it is still widely used in neuroscience, and it offers us a good way to discuss cytoarchitecture. As you can see on the right side, the Brodmann map is simply a way to divide the regions of the brain, but instead of doing it based on the different sulci and gyri, or based on the functions like we did, the divisions are based on the cytoarchitectures of the brain. In total, there are 52 divisions, and some of these Brodmann regions overlap with our structural and functional divisions, but for the most part, the map organizes the brain in a different way, although, as we will see shortly, it makes sense that there is an overlap between cytoarchitecture and function. An important concept that has been discovered within the field of cytoarchitecture is that the neurons that make up the cortex are arranged in the layered structure. From this, two main types of cortex have been established: the neocortex, which has 6 cellular layers or laminae, and the allocortex, which has 3 or 4 layers. The allocortex can be further subdivided into the archicortex and the paleocortex. Examples of the archicortex can be seen in the hippocampus, and for the paleocortex, it is mostly observed in the piriform cortex. Now, these two images of the different cortex that you see are the direct staining work of Brodmann himself. You will notice in the description of the neocortex that the staining comes from an adult human, whereas the allocortex staining comes from the hippocampus of a kangaroo. Regardless, the human brain also has allocortex in the hippocampal region, but it is interesting to see that Brodmann apparently worked with kangaroos, which is definitely an unexpected animal to be used for scientific purposes. Now, with the exception of the areas that I have mentioned that are made of allocortex, the rest of the brain is mostly made of neocortex, and for that reason, we will mostly focus on it to build our intuition on the cortex. So, if we take a closer look at the 6 layers of the neocortex, although at first glance it might be hard to distinguish the boundaries, you will notice that each layer has its respective differences based on the density, size, and shape of the cells. The first layer of the neocortex, which is situated closest to the skull, is also known as the molecular layer, and it is mostly composed out of scattered neurons, dendrites of neurons that are in the lower layers, and axons that project to these dendrites. The second layer is called the external granular cell layer and mostly contains dense levels of small spherical neurons. The third layer is called the external pyramidal cell layer and contains pyramidal neurons that progressively get bigger the deeper the layer goes. Then, we arrive at layer 4, the internal granular layer, which is a very important layer because it is the main recipient of sensory input from the thalamus, and as you will see soon, this layer is most prominent in sensory areas. Layer 4 is mostly made out of small spherical neurons called granule cells. The fifth layer, the internal pyramidal layer, also represents an important layer because it contains pyramidal neurons that constitute the main output pathway out of the neocortex. In regions that have very prominent layer 5s, like the primary motor area, the pyramidal neurons there can get very big. Finally, the last layer of the neocortex is called the multiform layer, and it is characterized by having a heterogeneous profile of neurons. Due to its proximity with white matter, this layer ends up blending with the incoming or outgoing axons. Now, although the neocortex has 6 layers everywhere it is represented, the relative size of each layer differs based on the cortical region. This idea is one of the primary considerations Brodmann took to make his map. If we consider how the neocortical layers in the primary visual cortex, or Brodmann Area 17, compare to the neocortical layers in the primary motor cortex, or Brodmann Area 4, you will see that they are strikingly different. The major difference between the two has to do with their layers 4 and 5. As we mentioned earlier, the layer 4 corresponds to the cortical layer that receives most of its inputs from the thalamus. As a result, since the primary visual cortex is a sensory area, it will have a prominent layer 4 to accommodate the input it receives from the thalamus. On the other hand, the primary motor cortex is more concerned with motor output, and so its layer 4 is pretty much non-existent, but its layer 5, which mediates the output, is very large in comparison to the primary visual cortex. One important point of terminology that is used to easily differentiate the type of cortex is whether or not the region has lots of granule cells. Remember that granule cells were located in layer 4, so areas like the primary visual cortex that have very prominent layer 4s will be referred to as granular cortex, whereas areas like the primary motor area that have close to no layer 4 will be referred to as agranular cortex. In other areas that are less specialized in their function and process a more integrative role, the relative cellular layers will be between sensory and motor areas. For example, this is area 5, which, as you can see, has a balanced layer 4 and 5 with respect to the 2 other areas we've covered. In terms of its function, area 5 is an area of the parietal lobe that is known for its involvement in somatosensory processing and many more complex cognitive functions. Hence, the main takeaway with this brief section on cytoarchitecture is to illustrate that the different properties and organizations of the neurons within the gray matter can help us determine or infer the function of the area that we are studying, which obviously in return helps us better understand the functional units of the brain. Now, obviously from this conversation, we haven't even scratched the surface of what is actually happening in the cortex because there are so many layers of complexity that make the study of the neocortex such an interesting but complex field. Many of the reasons why the study of the gray matter is so complex has to do with the properties of the neurons themselves. If you are familiar with my discussions on the properties of neurons in the central nervous system, you might recall that a fundamental role of the neurons there is to integrate signals from the different inputs they receive via synaptic summation mechanisms. As such, you can imagine that all of the cell bodies that you see on the left receive and give connections to nearby neurons in the horizontal and vertical axis. Some estimations believe that in the cortex, there are 16 billion neurons, so if each neuron produces thousands of synapses, the amount of connections in the brain rapidly becomes an insanely high number. Of course, the connections between the neurons are not done randomly, and there is a structure and a logic, but with the trillions of synapses that there are, the circuit rapidly gets messy, especially that it has to be studied on the micron scale. Another property of neurons that makes the circuit hard to keep track of is the fact that neurons can communicate in many ways through various neurotransmitters and receptors, which offers the neurons the capacity to excite, inhibit, and even modulate their postsynaptic partners. Because of that, there exist many different types of neurons that all differ on what signals they send and even differ on how they receive and interpret a given signal. One last aspect that I want to point out that makes the whole circuit even more complicated is the fact that the strength of the connections between the neurons can change through various mechanisms of plasticity, which again adds another level of complexity. Nevertheless, in future videos, we will revisit the gray matter to explore the model scientists have come up with to describe the different types of neurons, how they assembled, and most importantly, how they end up producing the cognitive functions we are all familiar with. With this information now in mind, we can conclude our discussion on the anatomy of the brain, or to be more precise, the forebrain. In summary of what we discussed, we have started our conversation by pointing out the principal frontiers and lobes of the brain, and then we discussed the general sulci and gyri of the lateral, medial, and ventral dorsal surface. With this anatomical terminology in mind, we then explored the functional anatomy of the brain, which gave us good insight as to how the brain is organized. Then, we briefly discussed the main internal and subcortical structures of the brain, and we concluded our conversation by pointing out what is going on in the neocortex. Our discussion on the anatomy of the forebrain overall concludes our discussion on the anatomy of the central nervous system, where we surveyed some important components and principles of the spinal cord, brainstem, and of course the forebrain. As you might be aware, there is much that remains to be discussed about the nervous system that we have yet to talk about. Notably, I briefly alluded to the complexities of the neocortex, and that is definitely a topic that we will need to consider in the future. In addition to that, we need to consider in more detail how the different sensory and motor systems operate at both the peripheral and central level, and we also have to consider how these systems integrate together to make more complex cognitive systems that allow us to think, speak, memorize, and so on. Furthermore, we need to consider the more precise anatomy of the different structures we have introduced in this video to better understand how they mediate their roles. We also need to consider the anatomy of the blood vessels that supply the central nervous system, as it is a critical aspect to take into account, especially when it comes to what can go wrong with the brain. Lastly, we need to consider how the delicate structures of the central nervous system are protected by the meninges and how the ventricles allow the production and the flow of cerebrospinal fluid. Thank you for watching this video. If there was anything unclear or there was a mistake somewhere in the video, make sure to let me know in the comment section. If you enjoyed this video and found it useful, you can consider leaving a like and subscribing to support the channel. On the right, you will see the informational resources that I've used to produce this video. Thank you again for watching, and I'll see you in our next discussion.